So one of the great pleasures and satisfactions of DPUK is seeing people come through and learn their trade to some extent and then go on to better things. There we have several examples of that. And I say I find it personally very satisfying. That begins with the early career researchers. And we had a fantastic day yesterday morning with those. And a big thank you to Sarah Bauermeister for organizing that and all the contributors that made it such a wonderful morning. And then we have the posters. And now it's my great pleasure to introduce the winners of the poster competition. We have 2 prizes. And this is in no particular order, okay? All right. And at this moment, we have the suspense that the television cameras are all holding for, and we look round at the different faces to see how they're getting on, and it drives me crazy, so I'm not going to prolong it anymore. So the winner of the Alzheimer's Research UK prize, Abdul Wafiq Ali from Swansea University. So Abdul is the winner of the Alzheimer's Research UK poster prize. I'm really, really pleased for him. And I just welcome Shona now to represent Alzheimer's UK and present it to him. No, we're done. We're done. That's fine. That's great. Okay, thank you. And the winner of the Novo Nordisk Prize, which will be presented by Dr. Omar Al-Masri on behalf of Novo Nordisk, is Dritri Ratra from the University of Oxford. Once again, thank you. Thank you. So before handing over to our afternoon program, I'd just like to draw your attention to these bits of paper. We may still have some left, but if you haven't grabbed one, then I do recommend it because it captures what we do in a very succinct way for you to take away for future reference. So thank you, Simon. Who is coming up now? Vanessa. Wonderful. Vanessa, over to you.
Thanks, everybody. It's getting to the end of the conference when people are just needing to be woken up to clap for the sessions, but thank you for that. Okay, so the session this afternoon is something very dear to my heart. We're going to be talking about how trials are moving, where they're moving, how they can be applicable for all. We've got some great speakers. But as I was thinking about how to introduce this, I found this paper and was reflecting on where we stand in terms of globalization for trials. And I think you can see there's a graph here from this paper from 2023 on the bottom left here. And it's really striking. There's obviously been a massive increase in clinical trials over the last few years in AD. And it's also very striking, actually, how much European trials are increasing compared with North America. But the reality is, if you look beyond North America or Europe, we're still looking at really small numbers. And a few statistics just to emphasize why this should be a concern for all of us. 2/3 of people living with Alzheimer's disease are in low and middle income countries. And if you look at the global costs that we attribute to Alzheimer's disease, it's massive, 1.3 trillion, not surprisingly going up even higher over the coming years. But most of those costs are driven by high income countries, and we probably underestimate the costs in low and middle income countries. That's partly because majority of the costs are around family costs. So it's a huge carer burden in low and middle income countries, and therefore almost certainly underestimated in terms of how you get those costs, but also in terms of identifying people who are actually living with Alzheimer's disease. So in this particular paper, they identified about 1,200 AD trials that were running, and only 11% roughly were in low and middle income countries. And what's interesting is when they talked about the limitations and why this was happening, so I've listed them here: lack of resources, low industry presence, regulatory obstacles and operational barriers. Actually, that sounds kind of familiar, doesn't it, really, sadly? But obviously a much bigger problem in these countries. And so why is this important for us? Well, I think we're all very aware that there are differences in terms of genetics, normal aging, risk of dementia, risk factors for dementia progressing in different countries. Certainly access to care, potentially treatment response to some new drugs, and level of adverse events. And of course, just access to education to understand why it may be important to come forward with early symptoms is going to be a big factor. And then fundamentally, healthcare access in terms of whether you're going for a biomarker test or you're interested in getting a biomarker test or accessing a disease-modifying treatment. And at this point, I would say that most trials continue to be designed for high-income countries. A lot of them still depend, obviously, on multimodal imaging and assessments and cognitive tests that are built around educated, high-income country populations. The other thing that is definitely a barrier is data. And the previous session was talking about data sharing, and that fundamentally is a problem and a concern and an issue and a barrier. But I would again say this is even more so in those countries. And if we're going to start building better AI models to predict who we need to be doing biomarkers on in order to understand who needs to access these treatments, it becomes an even bigger issue. Who is able to have that good data so we have good AI models? And the final point that I got from this paper was the fact that this obviously goes beyond AD. And I was really struck by this and actually wasn't aware. We think about in the UK and other high-income countries of vascular cognitive impairment being about 15-20% of the cases we see, but actually it's much higher in these countries as well. So there's this whole other cohort of people beyond AD that we need to be thinking about. Another thing I came across was the World Health Organization blueprint for dementia research that was published in 2022. And this, as you can imagine, had some generic aims in terms of the fact that we needed to broaden diversity in trials in dementia. Funding needed to be more evenly distributed, not only between countries, but also in terms of stage of disease and types of trials. And researchers should be more diverse as well in terms of their backgrounds and locations. But what struck me was that the World Health Organization had this list of calls in this blueprint. They thought we should be having a more inclusive database that not only looked at characteristics, but also the cost of dementia beyond high-income countries, which I think is something we should all be thinking about. Have an international network where we start to share data, but also techniques, training opportunities, and resources in terms of staffing, increase capacity to do trials, more trials in diverse settings, and thinking about risk reduction. And another big thing they talked about was empowering those with lived experience. And it just got me thinking again that we sit in our silos by and large in our individual research bases, certainly within our individual countries. And we're thinking about how we can work internationally. And certainly DPUK is thinking about how we can expand and work more widely. But this starts to become, I think, a really pressing issue when we're thinking about where we are with biomarkers and new treatments. And I just wanted to highlight that one great piece of work that Ben Underwood is doing at the moment that's supporting the readout study, but also the AD Smart platform that Paresh Malhotra is leading, is this new POPT platform, which is this great innovation where basically you're invited to comment on a PPIE question online. It's actually live now. At our TDF site meeting yesterday, Ben challenged us all to go and share this link, popt.org.uk, if you're interested. And you can go and comment on the PPI questions around, I think, AD Smart Paresh already. I'm looking at Paresh, he's nodding. So if you want to go and do that, do. But I think the bottom line is we're at a time of immense opportunity, and I'm sure we'll hear about some of this in the following talks. We've had the first 2 disease-modifying drugs for Alzheimer's approved in the last year. We're at groundbreaking times in terms of new biomarkers, including blood. And in the UK, we have a lot of extra resource going into dementia research by way of the Dementia Goals Programme and Neurodegeneration Initiative, and also things like the Dementia Trials Network that Cath Mummery leads. The Dementia Trials Accelerator, we have Cornelia van Dern here as well, who's leading that, and of course DPUK 3 renewal. But do we need to be thinking more about how we engage with countries and sites beyond our own. We need to be thinking about things being more accessible, diverse and generalizable. And I think enriching populations more than we do in trials. How do we get more efficient data sharing? How do we ensure sustainability? I know I spend a lot of time talking about this and saying, we think doing blood biomarker tests is simple. It's not simple in a lot of settings. So how do we make that more efficient? And how do we go beyond the UK? And do we need to rethink how we do things like PPIE? But I've talked enough at this point. I'm going to hand over to the speakers. We've got a great lineup this afternoon that's going to cover some of these topics in terms of where we are with clinical trials from Cath through to where we are with our first UK platform trial for dementia from Paresh, and then Charles Marshall talking about how we advance clinical trials in the UK. Sorry, that's Ruth Croney from Roche, but yeah, equitable representation for trials from Charles Marshall. So without further ado, I'm going to introduce Cath Mummery. Cath is a professor of neurology and consultant neurologist at the National Hospital in London. She's chair of the NIHR DTRC and director of the New Dementia Trials Network. So Cath, over to you.
Thanks, Vanessa. And it's such an important point that you make about the need for equality globally. And Jorge has done some amazing work trying to take this forward, and you showed his paper just there. But we're reaching a stage at the moment where we're actually getting more inequitable, not more equitable, with treatments in some countries and not others, trials in some countries and not others. I think it's critical that we focus on it. But anyway, hi, everybody. Thank you for inviting me, and it's a real pleasure to be here and to see you all. I'm Cath Mummery. I'm a clinician, first and foremost, and I'm a trialist, and I've been doing trials for about 20 odd years now, focusing primarily on early phase, but also in non-AD as well as AD, so in FTD and in LBD. Today, what I want to do is just give you a flavor of the sorts of things that are coming up, that are happening in early phase, in new clinical trials, because I think it's an incredibly exciting time. One of the reasons I say that is I can only give you a flavour. I can't tell you everything that's going on. I haven't got enough time. That wouldn't have been the case a few years back. So life is getting busier and busier in trials, and that's a great thing. These are my disclosures. Okay, so we have made, as Vanessa was just alluding to, we have made some incredible progress in the past years and months in terms of treatments. We do now have treatments that are fully approved, but we really are at the beginning of that treatment journey. We are at the foothills of where we want to get to. And if we want to get to those dizzying heights of halting Alzheimer's, or even, I dare say it, curing Alzheimer's, we've got an awful lot of work to do. This is my great-great-great-uncle. Looks a bit like a neurologist. He wasn't. He was a gentleman. In those days, you didn't tend to have to work if you had enough money in your family, and he liked climbing as a hobby. And he basically decided that what he was going to do, and he made a name for himself doing it in the late 19th century, was find peaks nobody else could climb and climb them. That was his motto. Basically, if you can't do it anyway. So he was quite a stubborn man and a persistent man, and he had great success. He was known as the father of modern mountaineering. But he wanted to take things a little bit further than just the European mountains that he put his name to. So he set his sights on this mountain, and you may know this one. This is Nanga Parbat. It's one of the highest and one of the hardest mountains to climb. It's known as the Killer Mountain. So he decided he was going to go up this with a team in 1895, and he never came down. Still up there somewhere. And so clearly that's not a success. He didn't reach the peak, but what he did do was he forged the way. He showed people the path so that people could then succeed and get to the peak, get to the top of that mountain. And for me, that is a really great inspiration for where we are in terms of treatments for Alzheimer's disease right now. We're at that beginning, and we need to keep going, and we will show the way for others to get to those peaks. I said I'm a clinician first and foremost, so I'm going to bring this back to patients because I think patients give us the questions that we need answers to. They also give us the answers if we listen to them. And this patient, that lady in the middle, Carol Jennings, she's probably altered the course of research in Alzheimer's disease more than most academics. She's an extraordinary woman. She was a teacher in Nottingham, and in the mid-'80s, she saw a notice saying that two researchers, Martin Rosser and John Hardy, were looking for families that were worried about dementia in their family. They were trying to find the causes for Alzheimer's disease. So she wrote this letter to them. And she basically said, look, there are loads of people in my family that are dying, and they're dying young, and I know that's not normal. I need you to look at them. And she persuaded every member of her family to have a blood test. The direct result of that was finding the first gene to cause Alzheimer's disease. And this is John Hardy, a very youthful John Hardy, back in the day. And so her intervention led to that discovery directly, which then led to the amyloid hypothesis, which then led to us focusing exclusively for some time on anti-amyloid therapies. We've now diversified a bit, but we really did go down that path. And that path has not been an easy ride, has it? We've had 20 years of negative trial after negative trial and a lot of debate as to whether or not this was the right route to go down. But we now have 2 fully approved drugs. We know that if you reduce amyloid enough in the brain, in mild Alzheimer's disease, you can cause cognitive slowing. But it's modest, and there are significant side effects. So we've got a beginning. Anti-amyloid immunotherapy is our beginning. It's a fantastic beginning. It's an incredible change in what we can do. We know we can change the disease. We didn't know that not so long ago. But it's the foundation. We need to do better. We need to do more. So where do we go from here? Interestingly, the results of those 2 pivotal trials on donanemab and lecanemab were predicted really accurately by a review paper that came out before they did. And this was written by Eric Karen and Bart de Strooper. And what they did was they looked at what they thought would be how low you needed to go with amyloid to make a clinical difference. And they predicted on their graph around 20 centiloids. They said, we think that will make a difference, then you'll slow down the course of the disease. This doesn't normally happen, but that prediction became the truth when you look at all of the anti-amyloid immunotherapies. On the right, what you see here is the negative trials, and on the left, you see the positive trials. Negative trials did not lower amyloid low enough. Positive trials all lowered amyloid down to 25 centiloids or below. So we start to understand what we need to do to make these drugs work. We need to get amyloid down low enough, and we also saw in the trials, at least, you need to get it down fast enough. If you don't get it down fast enough, you're not going to spend long enough with it low to see an effect in a trial setting. So how do we do that? How do we accelerate amyloid removal, get it down quickly, but not increase those side effects that everybody worries about so much? For many years, we've been happy to give a drug in huge concentrations so that we get a tiny concentration across the blood-brain barrier. 0.1% of the drug gets across. That's ridiculous. Why haven't we been doing something about that? So we've been doing that up until now. Active transport mechanisms are something that many different companies are looking at and developing, and this was one of the earliest ones by Roche. So what they did was they took the transferrin receptor and they adapted an anti-amyloid immunotherapy so that it used the transferrin receptor as a mechanism to transport it across the blood-brain barrier. And if you do that, you are far more efficient at getting drug in. Of course, the question is, does that cause more side effects or problems? And currently, the phase 1b trial is ongoing. So these results are from a study that's happening right now. And what you see is that amyloid is dropped. If you see that purple bottom line, that's the top dose. Amyloid is dropped by 12 weeks, so they've had 2 doses, by 89 centiloids. Now, that's a standard measure of amyloid. That's more than lecanemab dropped it in 18 months, and it's about the same as donanemab in 18 months. So that's a huge acceleration in the ability to reduce amyloid in the brain. And associated with that you do not see an increase in ARIA in the side effects. You see a significant reduction. You see very few ARIA because the drug gets into the system in a different way, and therefore it's much safer. So this is early days. Obviously, you need far bigger trials to be done to validate this, and we need to see cognitive change. But I think these active transport mechanisms are going to be something that really change the game for many of our biologics, our large molecules. Very exciting, I think. But do we think taking out amyloid is enough? If you look at post-mortem data of people that have been treated with anti-amyloid therapies, it gives us some clues as to whether that's true or not. So if you look at this slide, on the right, that's an individual that was given aducanumab. On the left is a case series of people that had AN1792, which was an active vaccine against amyloid many years ago. That study was stopped prematurely for reasons we won't go into. But basically, what you see in all of these people is a clear reduction in levels of amyloid. The amyloid all looks moth-eaten. It's surrounded by activated microglia who are busy phagocytosing it. And you do see a lowering of accumulation of tau, but they all progressed clinically. And when you got to post-mortem, they all had widespread tau. So even though you take out the amyloid, certainly at these stages, you still see tau accumulation and disease progression. So either we have to treat really early before any tau accumulates, or we need combination therapies. And I'll come back to that point in a second.
An event seen from one point of view gives one impression. Seen from another point of view, it gives quite a different impression. But it's only when you get the whole picture you can fully understand what's going on.
So I really like that video. It just makes sense to me. We need to look at the whole picture. We have focused on amyloid for a very long time, but it's a complex disease. We know it's a complex disease. We know there are multiple things that we can target in Alzheimer's disease. And I think it's critical to optimize our ability to treat successfully, to look at multiple things. So just looking at amyloid, but in a different way. At the moment, what I've talked about is mopping up amyloid from the brain, using monoclonal antibodies to try and remove the amyloid that's already there. But why don't we work upstream of that? Why don't we try and decrease production of amyloid, especially in people where we think there might be an overproduction, and try and therefore decrease that accumulation? And you can do that with gene silencing. So these methods, and this particular one uses an interference RNA, which is called mivelsiran, developed by Alnylam. And it targets the gene and the way that the gene is transcribed and then translated into the protein. And what it does is it binds to the cell machinery. It hijacks the cell's normal machinery that regulates the gene production, and then prevents that message from the gene, the mRNA, from being translated into the protein. So it reduces levels of the protein, in all compartments and all of the products of the protein, including APP fragments, which lead to amyloid development. And so this is a study, again, that's a phase 1 first-in-human study that's ongoing. And we've completed the single ascending dose part of this study, where everybody has one dose. That's what I'm going to show you here. Now they're getting multiple doses to see what happens next. So this is a gene therapy or a gene silencing therapy. It has to be given intrathecally because it's a large molecule. Can't get into CNS otherwise. And this is data from one dose. So these individuals have had one dose only at different doses, as you see there. They're young onset Alzheimer's disease patients because we think they have greater production than impaired clearance. So the rationale there is that we might do them some good. This is a phase 1 study, so safety first is the primary endpoint. And then we also look at whether we can see changes in the fragments of APP in the spinal fluid. And these are the initial results. So the study is still ongoing, I hasten to add. But if you look at this, we've got data out to 12 months across those different doses. And what you see is, bar the lowest dose, a really dramatic reduction in fragments of APP in the spinal fluid. And not only is it dramatic, it's down to about 80% reduction from baseline, it's sustained out to 12 months, one dose. So think about the difference between having Leqembi every 2 weeks as an infusion and 1 dose intrathecally once a year. It makes a big difference. And what we see is that's reflected in the precursors of amyloid plaques as well. So we can see reductions in A-beta-42 and reductions in A-beta-40. Now, the important thing here is, and so far this has been safe, one thing I would say is there are a lot of lumbar punctures in this trial, and what we see in terms of side effects, basically the side effects that you have from having a lumbar puncture, back pain or headache. Otherwise, there have been no SAEs related to drug, and there's no ARIA, because you're not removing amyloid. It's a different mechanism, and that means the populations that could be at risk from anti-amyloid immunotherapies could be safe with this different way of trying to change amyloid. So we wait and see what we see with the next steps, and we're currently dosing multi-doses every 6 to 9 months to see how that goes. Enough about amyloid. I mentioned tau, and I think it's critical that we focus on tau and how we can change the course of that. And we know that it's got a role, obviously, but we also know that it's different to what happens with amyloid. By the time you have symptoms, you have a head full of the stuff, head full of amyloid. But as your symptoms develop, your tau pathology and spread and severity correlates really closely. So it makes sense to think about targeting it. We also know, although it's intracellular, it seeds between cells. So in that part of it, it's extracellular. And we know that the tau-amyloid interaction is bidirectional. So it's not just amyloid leading to tau. Tau also then has a reciprocal impact on amyloid toxicity. So basically, if we can reduce it, we can hopefully reduce that spread, the aggregation, try and reduce that toxicity. I used to have, not so long ago, at the bottom of this slide, that this is a later treatment option. It probably is a little bit later than amyloid treatments, but I don't think it's a late treatment option. And the reason I say that is because of this sort of data. So this slide is 3 graphs, one from the medial temporal lobe on the left, middle is temporal cortex, and this is rest of the cortex on the right. And on the x-axis is your level of amyloid on the brain. On the y-axis is whether you're tau PET positive or negative. And what you see is up until around 60 centiloids, there's not very many people that are tau PET positive. And then there is a sudden explosion. And way more people at 60 centiloids have become tau PET positive, in particular in the medial temporal lobe. So what does that tell you? Well, it suggests that the amyloid accumulates, and then to a point, it bothers tau. And when it bothers tau, it's like dropping a match in a forest fire, right? The whole thing accelerates rapidly. So to me, that means we should be really thinking about these tau treatments around 60 centiloids or before, but you don't have symptoms then yet. So when I said later, it's definitely not late. And we have to think about this when we're thinking about our diagnoses, our biomarkers, our biological tests. 2 examples of tau therapies I just briefly want to mention. One is tau immunotherapy. So like the amyloid immunotherapies, this is a monoclonal antibody that's given as an infusion, and it tries to mop up the tau as it's traveling between the cells, seeding between the cells. Every trial has been negative so far, with one possible early exception that was recently shown at one of the conferences. And the thinking behind that is that that's possibly because they've been targeting the wrong bit of tau, not the pathological stuff that seeds between the cells. And so Rohan de Silva at UCL, with Eisai, in a really great collaboration, developed E2814. And this is a tau immunotherapy with a bit of a difference. It targets the pathological tau, the bit that seeds between the cells. And we started a study in 2021 in people with autosomal dominant Alzheimer's disease using this drug. Now, this is a tiny study. Although it's an international study, it has 7 people in it. That's absolutely minute. The aim was, as a proof of concept, to show that we could change things to put it into a bigger study. And we finished the study last year. And what we saw was, firstly, it was safe. There was no safety signal with the drug, which is a good start. And these are 7 people, so there are no statistics here. This is just observational, right? What we saw was that in these individuals, in the spinal fluid, the levels of tau did seem to decrease. They varied depending on the people, and one of them seems to have some resistance, but they did, in majority of people, they seemed to decrease. And interestingly, it was a form of tau that correlates with the tau tangles in the brain. So the suggestion is this might be disease modification, but in 7 people, you've really got to be cautious about that. So we're putting it into a much bigger study, which is currently running, in those at risk of autosomal dominant Alzheimer's disease. So in that earlier population before symptoms. And look how the world has changed. So now this trial is giving that drug, but it's giving it in combination with lecanemab. So this is the first trial to give an anti-amyloid therapy as well as an anti-tau therapy. And you can imagine that adds layers of complexity to your safety, to your results, etc. But that is the direction that we are going in terms of future trials. And we wait and see what this shows when it reads out. The second tau drug I wanted to mention is coming back to that idea of gene silencing, trying to reduce production of these toxic proteins rather than try and mop them up once they have done their damage. So this is a similar process, but not the same as the interference RNA. This is an antisense oligonucleotide. So what this does is synthetic oligonucleotide. It binds to the messenger RNA that comes from the gene. So the MAPT gene produces a messenger RNA, which is then translated into tau protein. In this case, BIB80, this drug, binds directly to the message, causes it to be degraded by the cell's machinery, and therefore decreases tau production. In mice, what that showed in red versus dark gray is if you give drug to mice that accumulate too much tau, you see a reduction in tau accumulation, you see better survival, and you see normalization of nesting behavior, which is a behavioral test that they use in the mice. But importantly, you don't just see that slowing of accumulation. You actually see in the aged mice at the bottom a reduction in the levels of tau in the brain, which suggests they've reversed the tau burden in the brain. And that was something that wasn't actually expected. It was better than they expected. So it went into a clinical trial in 2017, back in the day, which was quite scary, giving intrathecal drugs to people in gene silencing in 2017. We all thought we were mad. And this was a multiple dose ascending study in mild Alzheimer's disease. The study started 2017, finished 2022. And you hear how long these trials are. They're only in there for a period of 12 months, 18 months, sorry, including the extension. But because of the recruitment rate, it takes a long time. And what you had was increasing cohort doses, initially monthly dosing, again intrathecally, and then by the 4th cohort, every 3 months. They then went into a long-term extension if they wanted to, and every single person did. And we measured levels of tau in the spinal fluid every time they had a dose. And in the last 2 cohorts, we measured what happened on their tau PET scans. And when you look at the CSF and you look at the spinal fluid, what you see is that the MAD is the placebo-controlled part of the trial, and you see a dose-dependent reduction in the levels of tau in the spinal fluid, which is terrific, down to around 40% of baseline. When everybody gets drug, that converges, including the placebo groups, then also show that reduction down to around 40%. So really encouraging, but what does that actually mean in the brain? This is different. It's a dynamic process in the CSF. Small numbers. We need to see bigger numbers to validate this. But what we saw suggested that across all of the groups in the extension studies, so everybody's getting drug, in every group, across every part of the brain, there was a lowering of tau in the brain, not a slowing of accumulation, a lowering. If you look at 2 examples of what that looked like, 2 individuals on the left-hand side at screening, in the middle, they have had placebo for a period of time, 6 months, and you see that the tau accumulation is at least as much as it was, maybe a little bit more than at screening. On the right, they've been on drug, and it's clearly lower than it was, and that was the same in all of the individuals in the study. So a bigger study is ongoing. We will wait and see what happens when you do this in 370 people, not in 46. But I think, again, this is the first time we've shown that you can change tau in the brain, as the mouse model actually suggested. Intrathecal injections once a year aren't that bad, but we'd like to be able to give things in a less burdensome way. And I mentioned active transport mechanisms at the beginning of the talk with the brain shuttles trial. Well, why can't we do that with these drugs as well? And that's exactly what we're planning to do. Denali have, like Roche, developed an active transport mechanism. And what they're doing is binding that to an antisense oligonucleotide so that you can give it intravenously and not intrathecally, which will make a big difference. What you see here on the left is a monkey brain, and this monkey has been injected with the intrathecal version. And you see how bright it is in the spinal cord. That's because when you inject here huge concentrations around the spinal cord, and you then get a gradient up to the head. And in the head, you see patchy distribution and nothing in the deep structures of the brain. This is what it looks like if you give it via this active transport mechanism. Nothing in the spinal cord, because it's being given intravenously. But because it goes through this such a vascularized process in the brain, and it's via the choroplexus rather than the rest of the system, you see this homogeneous ribboning of the cortex, and it's getting to the deep structures. It's completely different. So again, delivery changes are really going to help us with these therapies. The last drug mechanism I want to talk about very briefly, because I think this is super important, is the immune system and how the immune system impacts Alzheimer's and other neurodegenerative diseases. We know from the genetics that immune-important genes, such as ApoE4, such as TREM2, are critical risk genes in Alzheimer's. So we know it's important. And if you look, and you've all seen this graph, I don't know how many times, but if you look at the Jack curves of increases in amyloid, followed by tau, followed by neurodegeneration, all when people are still normal, no symptoms, and you put on the immune system and its responses, there are changes very early on in the course of Alzheimer's disease. And this suggests that maybe the immune response isn't just a response to the proteins triggering it. Maybe it's part of the driver of the process in the first place, and we really need to think about how we treat it. And so there are several mechanisms that are being used. One has been borrowed from cancer. So in cancer, checkpoint inhibitors have revolutionized the way that cancer is treated, using the immune system, enhancing its function so that it can target the cancer cells. And Michael Schwartz has worked for 25 years on how the peripheral system, the immune system, and the brain interact and how one might impact the other. And as a result of that work, this checkpoint inhibitor was developed targeting neurodegeneration in the brain, including Alzheimer's disease. And the aim here is that you enhance the peripheral immune system, you reverse the senescence that happens as we get older, you enable it to work better, which leads to more trafficking of specialized cells into the brain, which leads to better clearance of proteins and better immune processing in the brain, and therefore, hopefully, slowing of cognitive decline. The animal models suggest that that's what it does. We're currently doing the phase 1 trial using this drug. Now, because this is such a different methodology, we were quite trepidatious about the idea of checkpoint inhibition in Alzheimer's. And we brought in a lot of oncologists who have huge experience in giving these drugs in cancer patients, who have been really reassuring in holding our hands throughout the whole process of this trial. But the trial is now fully recruited. We've completed all of the single ascending dose, the blue, and we're now completing the multiple ascending part of it. There have been no drug-related serious adverse events. There have been a few immune-related events, mainly asymptomatic thyroiditis that resolves when you pause the drug. So far, again, well tolerated, and we need to watch this space to see what this looks like. So what do I think we're doing with all these drugs? I think we're building a portfolio. We're building an armamentarium of things that we can use in individuals depending on where they are in their disease and what their biomarker profile looks like. I think we're working towards, it's a way off, but we're working towards a tailored approach to each individual that gives them the best drugs for where they are and what they have in terms of their disease. Just briefly before I finish, I want to just pivot to tell you how I think it's so important that we look at the global picture, as Vanessa was talking about, that we work together to really expand what we can do across the UK and make the UK a go-to place for doing these things. This is a map of phase 1b trials in Alzheimer's disease. Looks like the States do everything. If you drill down into it, that's not quite the case. Currently, UK outperforms in terms of number of phase 1b trials done than anywhere else other than a few states in the US. That wasn't the case 10 years ago. We've managed to change that over the past 10 years. We've managed to show we can do it. Companies will come here, which is terrific. But we need to be able to do more of it and better, and we need to build capacity and expertise across the country. And that's the aim, of the UK Dementia Trials Network, and Vanessa kindly gave me an intro to that earlier. And this was funded by the government last year. Basically, the aim here is get 20 sites to a really expert level across the UK to be able to do these early phase trials, to really accelerate innovation and the sorts of things I've been talking to you about. And this network is just being built at the moment. There are many people in the room that have been involved in those discussions. Vanessa, as my deputy chair for the Dementia Translational Research Collaboration, is very involved in this and the wider work. And the aim here really is to work with industry and with sites and make sure that we accelerate what we're doing and improve participant support as well. So there's a number of sites you see there. Those are the sites that have applied to be part of the network, and 32 of them in total. We're just finalizing the first wave of sites, but we're also talking to all of the other sites in terms of how we can bring them on and what they need to do that. And we're developing a real-time feasibility dashboard that shows exactly what each site can and can't do at any particular time. The pilot has been ongoing with 3 pilot sites starred there, and we've started our first phase 1b trial, which is a gene silencing therapy. We started at the beginning of this year. The first patient, please keep your fingers crossed, will be dosed on Tuesday at one of our sites, which is really exciting because that's the first dosing in the network. And so far, pharma have been really encouraged by the way we've been able to communicate, speed things up with contracts, and collaborate in different ways, whether it's networking across imaging or making sure that the sites are just all talking to each other. We all know if you do trials that actually all too often people are in silos. All we have to do is talk more often. It's not rocket science. Industry engagement is a very important part of this network, and I won't go into this in detail, but just to highlight a couple of things. Firstly, ensuring that the whole community, the wider landscape in the UK talk together, and Vanessa again mentioned this, the Industry Liaison Forum is something that we have developed alongside Paresh at the RDN, Vanessa at the TDF, to really show, and the wider CTA, I think, DTA, I hear, yes, you are. And the Dementia Mission as was, which is the Goals Programme, to really show that we are all talking together to make sure the UK has a pathway to success, as JP would put it. The feedback, by the way, is that right now is the best possible time for us to be developing things in dementia, in trials. The US is in an interesting position, as we all know. Europe is being increasingly conservative, and so there is an opportunity for us to really build things here, and I think that is a very real opportunity. The last thing I want to mention on this is the collaborative projects are really a big, important part of this network. And I know that Ruth is going to talk later, and she'll give a couple of really nice examples of how Roche in particular, but also others, have joined together with the different initiatives to try and improve different parts of the landscape. So the vision is increase capacity and capability, make it faster, make it high quality, better access for everybody regardless of where they live and who they are, developing trials leaders and trials career pathways, and really working as a network alongside industry and the rest of the infrastructure. So I started with Carol. We thought we'd have a treatment. John told me he thought we'd have a treatment 5 years after that particular paper was published. Unfortunately, it's taken us a little bit longer than that. We didn't have a treatment in time for Carol. She died last year, unfortunately, but her children we will have treatments for, and we will have bigger and better treatments for. And so I think that's a huge incentive for us to continue this work. And this just reminds me that dementia is quite a dry, unappealing word. These are all participants in current trials that I'm currently doing, and they all have, you can just see on their faces, they all have rich, enjoyable lives that we need to try and help them with. And just want to thank all of them and all of the others for their commitment and their time to what we do, because we wouldn't have any of this work without them. Thank you for listening. Thank you to my team, and thank you particularly to the patients.
Thanks very much, Cath. And I think we've got time for a couple of questions. For Cath, we're going to have a panel at the end.
Thank you very much for that talk. Totally in awe of the amazing work and in particular how you keep it all in your head to present it so clearly. However, I'm a public health doctor, so my question when I come to this field is how we reduce the morbidity by as much as possible. And I think there's two kind of key questions that come out of this for that perspective. The first is, it's always concerning when we talk about having to do things to people who don't have symptoms. So I totally understand the rationale, but my worry is how we find those people. So we've got these prediction models, Clifford Jack has one, and they have lifetime risk for people who have asymptomatic amyloidosis of less than 50%, way less than 50% for most people. And so you have this paradox of if you wait till the neurodegeneration starts, then you have higher lifetime risk and we justify more things. But of course, always saying from the data is that we need to go sooner. So how do we actually find these people? And then the flip side is that the trials are really, really selective of who's in them, but perfectly justifiable efficacy reasons. But I think the lecanemab trial had an average age of about 71. In the CFAS data for epidemiology, we have median age of onset of symptoms of dementia about 83, or incident dementia diagnosis. So you have this massive gap. And it's all well and good saying we're going to give people multiple therapies, but we know lots about polypharmacy and people in their 80s. And so how do we try and bridge those two things of selection of people that haven't got symptoms and actually treating the population experience of disease?
Really good questions. I think, have I got about an hour to answer that question? Or 3 questions? So, look, I think they're both highly relevant and important points. To the first one, in terms of asymptomatic, we're in a really interesting place right now with Alzheimer's disease. We have biomarkers, unlike a lot of the other neurodegeneration, and they're improving very quickly. We are able now beginning to, and studies like READOUT and ADAPT are going to be helping with this, validate those in the real world. Those are in symptomatic people. We are able to use them screening in research, and that's very successful. But how do we predict when somebody's positive on a blood biomarker when they're going to get disease? At the moment, we can't clearly, but we are beginning to see from work like Oscar Hansen's and the Biofinder work that there are signs that we can start predicting. We're not there yet. So any prevention studies I'm doing are either in people where you've got proven amyloid, and it's a very long study in sporadic. Now, that's mainly Risa Sperling in the States, but it's very interesting work, and look at that if you're interested. I do a lot of prevention work in genetic Alzheimer's disease. And you start there where you know when somebody's going to get disease, you know when things are going to change. It's a much simpler model from that point of view. It's still not easy, but you know what's going to happen. So, and how you find those people, again, what's changing at the moment, and there's a big debate, as you know, in the community about purely biological diagnosis versus biological and clinical diagnosis. Some people are right over here with we should be making a purely biological diagnosis. I don't think we're there yet. I think because of the point you make about the difficulty with predicting when things happen, we're at that kind of slightly uncomfortable midpoint where we know we're going to be going there at some point, but we haven't got all of the the weapons that we need to do it yet. So that's my answer to your first point. Your second one about these being really selective. Yep. Absolutely agree completely. These are very well fit people with Alzheimer's disease. You exclude most of the ones that have any significant vascular, and that gives you an answer, but it doesn't give you an answer for the majority of the population that have combined pathology or have comorbidities or can't have it because they've got uncontrolled hypertension. We haven't been in a position where we've been able to do real world studies until very recently because we need to see something to then take that forwards. So I think what you're talking about is going to be a huge wave of work ongoing in terms of real world trials with people that do have, and Paresh is going to be talking later and may well mention his thoughts on this, which I think are highly relevant because he's very carefully being inclusive in his trial. So firstly, we need to be more inclusive. I think there's so many other things we need to be doing there, by the way. We're not even looking at male versus female. We can't tell you what it means to be Afro-Caribbean and have lecanemab, donanemab, or any of the others. There's a huge amount of work to be done. But it's the beginning, like I said, right? There's so much we need to do. So we need to get busy.
Okay, Michelle.
Hi, Cath. Great talk.
Thank you.
I don't think we need to worry too much. What if I could offer you, and this is not theoretical, a well-phenotyped group of patients in mid-60s for whom we know they have a 6% per year chance of a future conversion to dementia with Lewy body or Parkinson's, who also have evidence for Alzheimer co-pathology, which becomes increasingly relevant to the DLB group. It can be identified by blood-based biomarkers prior to conversion. What would your thoughts be on doing a trial on this patient group that might need, dual therapy? 1, alpha-synuclein, plus or minus 2, amyloid beta.
Very good question. Actually, Ramona asked me that question last week.
Did she?
So we're talking about it next week. I think it's... we can get together. Listen, I think it's really important that you have a well-defined preclinical population. It's what we're talking about, with predictions. We still need biomarkers for outcome measures. I think that's perhaps less well-developed than it is in terms of Alzheimer's disease, but we can discuss. I have no issue whatsoever with trying to deal with 2 proteinopathies at a time, because I think that's where we're going to be going. We're basically going to be sitting there saying, well, this person has on their biomarker profile these proteinopathies. This is what we need to give them. I hope we will be less label-based at that point and more about the pathology and what it looks like and how to treat it. One caveat that this is one of those hurdles that we're really going to need to try and understand better. The anti-amyloid immunotherapies and the post-mortems, there's one that I hadn't got on there because I couldn't find a picture, which is where somebody that had co-pathology with significant amyloid and significant vascular was given anti-amyloid immunotherapy. It cleared the amyloid, but not as well. And they had far less benefit than the others. And I think that the interactions amongst all of these proteins are something, again, there's so much work that we've got to do. This is just the start. But yeah, let's talk.
Lovely. And we will have a panel at the end, so if you have more questions for Cath, we can ask more later. Okay, thank you again, Cath.
So not only do I get Friday afternoon, I get Friday afternoon after Cath Mummery. So I apologize in advance. I have no data whatsoever to show you. I have very little work that I've done myself, and mainly it's a talk about people and trying to bring them together. So this is my list of roles and conflicts of interest. The most important one, I think, and I think Cath highlighted this too, is number 3, as a consultant neurologist. This is the beautiful building where I practice, which is Charing Cross Hospital in West London. And every Monday morning, I see patients there. And although we hear about the great advances that have taken place with anti-amyloid immunotherapies, my options for treatment are from 2 or 3 decades ago. One which was literally being developed when I was a medical student. So we haven't really got very far. We hear a lot about these medications, and there are massive advances, as I mentioned, but they are for selected patients who do not have co-pathologies, and our colleagues in pharma are going earlier and earlier in the course of disease with good reason and with a good rationale. So what are we going to do for patients who we see in clinic at the moment and who have clear dementia and mild cognitive impairment? I put a grant application in before Donald Trump's first presidency for a repurposing study. And we heard about repurposing from Jodie yesterday, and I think we've heard about it a bit in other talks as well. And this was a repurposing study with a company that doesn't exist anymore, Shire, which has been taken over by Takeda, to look at a drug called guanfacine, which is a drug used in ADHD, to see if we could combine noradrenergic therapy with cholinergic therapy and see if it makes a difference to patients' symptoms. And I'm not going to talk at all about the results of that. It's taken a long time to finish it, and we finished it at the end of last year. What I'm going to talk about is the fact that it's done now, and all the team, some of whom are in the room, is Cara in the room? I can't see. So Cara was there at the end to finish it all off. The team disband, the study's over, and we take the stadium down. So we build a stadium for one trial, we play our one football match, and then we take it all down, and we have to do it again. And these are the trials that are taking place worldwide for Alzheimer's at the moment, and almost all of them work on that basis. Let's set the team up, let's play one game, and then take it all down again. I appreciate I'm mixing my metaphor as I go, but nonetheless, it's a very inefficient way of doing things, this sort of comparing a treatment with standard of care and then stopping and doing everything in a very serial way. So Cath showed the DIAN-TU study, which is a platform trial in familial Alzheimer's disease. And I think Tim this morning mentioned us being 30 years behind cancer. In this case, we're almost exactly 20 years behind cancer. This is Max Palmer, who's a professor at the MRC Clinical Trials Unit at UCL. This is the STAMPEDE trial for prostate cancer, which he was part of setting up in 2005. And it's a platform study, a rolling study of treatments for prostate cancer where you get diagnosed and arms are set up and got rid of if they don't work, and then new arms come out as they are developed. So you have a rolling trial for testing treatments. You are not taking the stadium down after every match, but you are ensuring that your team still exists and that really you're being as efficient as possible. So part of the problem with the NORAD trial, which I showed you earlier, is my deep attachment to it. I, as an investigator, want it to work. And that applies to most investigators. But rather than being like that, we should be agnostic about our treatments and fail fast if they're not going to work, so that we can get on with the next one. And STAMPEDE finished in 2023 with 4 changes of practice because of it, and STAMPEDE 2 is about to start. So this idea of a platform has worked in cancer, and has also, as well as DIAN-TU, come to neurodegenerative disease as well. So this is the MND SMART trial run by Suvanka Pal out of Edinburgh, looking at repurposed medications, in this case memantine and trazodone, comparing with placebo in motor neuron disease. And that's recruited about 1,000 patients at 20-odd sites in the UK. And this is the Octopus trial run by Jeremy Chataway at UCLH, looking at progressive MS. So we have experience in other diseases, and we have experience in other neurodegenerative diseases. And so a group of people, including Suvanka, including Jeremy, set up this group called Accord, which is a group, well, it says what it is, but it's a group of people trying to set up these platforms in neurodegenerative disease. And as you'll see, you need a group of people. You can't have one person putting in a grant 10 years ago and slowly trudging through every process. It requires a massive team and people with expertise across the board to get this sort of thing going, and it requires support from charities and from institutions, all working together to try and achieve what we're trying to do. And so we've been talking about setting up a platform in Alzheimer's disease for probably 2 or 3 years now, and have been galvanized by various grant calls and by the support of charities. And for the last couple of years, primarily a partnership between Accord and DPUK, we've started to plan this out. And we want to repurpose in the first phase. And speaking to the points from the last talk and the discussion, we want this to be an inclusive trial for the people that I see in clinic on a Monday morning. The people with hypertension, the people with diabetes, the people who have small vessel disease on their scan, rather than the purer, more selective population that we've been talking about. And we want them to have symptoms. So an MMSE of over 17. And so for those of us who think in terms of MMSEs, that's impaired. It's not terribly impaired, and it means that they should be able to take part. But it does mean people are impaired. And we're looking for a sustained clinical response over 18 months, and we want to use validated clinical outcomes. And we'll use scans and we'll use blood-based biomarkers, but we want to know that these things have made a difference to people and people's lives. And here you can see on the right our initial team of people, some of whom are in the room, some of whom no longer have their beards. It's mainly come about because of the enthusiasm and the contribution of all these people. So because they're there, and these people here are taking part and giving a lot of effort to this, we're slowly getting somewhere. Just to give you an idea of the design, So what we're talking about roughly is around 300 people per arm, starting with 2 arms and comparing those with standard of care, and then starting a 3rd arm as we get to about 18 to 24 months. And as I said about failing fast, you have preliminary analysis stages before you get to your 18-month primary outcome. So you have a preliminary analysis at 6 months and another at 12 months. If something looks like it's not working at all, or, which is possible, making people worse, you stop, and then you start something else. So you don't persevere and persevere because it was your idea, but you get on with the next thing. And so the best bit about preparing these talks, you can tell I'm not very good at preparing slides because it's all very messily done. But the best bit is adding people's photos as more people come on board. And we still don't have enough people to deliver this work. So these are our core leads who have been setting things up. We have an application development award from NIHR, which has helped us get things started. And I'm just going to go through the aspects of that, but that's the drug prioritization pipeline led by Suzanne Reeves from UCL, thinking about our outcome measures led by Ross Dunn in Manchester, design finalization led by James Carpenter from the MRC CTU, and PPIE, which is critical, and I keep saying it last, I'm going to start saying it first next time, which is led by Ben Underwood, whom you've heard a bit about, and Linda Poynton, who has experience of dementia in her family as well, who works in Cambridge. So we're meeting weekly to try and set up and design this study. And so the first bit was choosing what we're going to give people. And we put out an invitation to the general community for proposals for drugs last summer now, and we got together this stellar panel, at least one of whom is in the room, and we also asked people to propose their drugs to begin with, and I think at least one proposer is in the room as well. And this panel gave their time and effort, some of them waking up at 5 in the morning, West Coast time, to listen to people's proposals, go through extended drug CVs, and then rank compounds. And then they went through even more information at a second panel meeting to rank again with more information so that they could choose the drugs that we would work with. And so we had 14 compounds presented in the first meeting, 10 shortlisted, then a very busy time for some other people in the room, scoping the drugs, preparing the drug CVs, which were based on the MND SMART work, so that we could use some of the mechanisms that have been set up for that. Separate weekly PPIE meetings to discuss the drugs in detail and give the information to our PPIE group, and then second panel and selection in December 2024. So now we know that we have metformin and atomoxetine as our first drugs to start with. We have our design, as I've shown you already, with the 18-month outcome, and we know still that we're randomizing roughly 300 people per arm at this stage. And we've met with MHRA, and they're very happy with our design. They want us to think about having a co-primary outcome measure as well. So our outcome measures, as I said, were validated clinical outcomes using the ADAS-COG as our primary. But what MHRA want us to do is think about having a co-primary using an activities of daily living score, and we will probably go with the Amsterdam ADL score. Roche is thinking about all the aspects of outcome measures and when we actually assess patients. But one of the other things that's come up, and that's come up in Michelle's talk, but many other talks today, is our use of digital outcome measures. And these bars show the use of digital outcome measures in trials, with the yellowy orange color being Alzheimer's disease studies. And everybody has a digital outcome measure that they are interested in or they think that works. And many of them are cool. I think lots of them are cool. But whether patients and their families will think it is cool to be doing 5 or 6 digital outcome measures as well as the validated clinical outcomes is not so sure. So what we need to do is have a separate panel with also some people in the room looking at the different digital outcome measures that we could use, and that will actually add to the information that we get from our primary validated outcome measures. So MHRA, thankfully, were very clear about this, that you use things that will help you work out whether a drug works or not. That's what you incorporate. And so we check with our PPIE group, and that's how we'll choose which digital outcome measures go with the validated clinical ones. And so anybody who has a digital outcome measure that they would like to push, start preparing to email Ross, who will be asking for proposals very soon. As I said, PPIE, led by Ben and Linda, should be in every area of the trial. People are sitting in on the drug selection panel, sitting in on the outcome selection panel, also discussing the design. Everything is done according to the proposed framework. But as Vanessa has already shown you, we want to do that a bit differently too. So this is the website that Ben has produced called POP. You've heard a bit about it. I am not changing slides unless you get your phones out and take a picture of my QR code, because that is the only thing I have done all week, is to prepare this QR code, and it took me about an hour in Safari. So I'd be very grateful if some of you can get your phones out, take a picture, and fill in the survey when you get the chance. But this sort of approach, getting PPIE at scale, is what we need to be doing. And if we can marry it with things like JDR and other web-based approaches to how we recruit and how we think about trials, we will get further and we will get there faster as well. So I'm indebted to Ben and Linda and Zanira, who works with the Care Network at KCL, sort of trying to increase the diversity of our PPIE group, for really pushing this aspect in every part of what we're trying to set up and making us think not just about racial diversity, but also thinking about the traveller community, thinking about people who don't have very good hearing, and how we make sure that as many people as possible get the chance to take part in research and take part in this trial. So just about the last slide, is just to say that our trial is going to be called AD Smart. A lot of what we are trying to do is based on the O'Shaughnessy report and the government's response to it, which, if you haven't read, I would strongly recommend you read, because although it says that it's about commercial clinical trials, I think it applies to everything that we do and the research that we carry out with patients. It's basically about doing more for patients and their families and making it easier for them to participate in trials. And that's very much what we want to be. We will start off with repurposed medications, but like Stampede, like the Healy motor neuron disease trial in the US, we will be open to having industry-sponsored arms. We want to be participant-friendly, and things are moving. Things are moving. You've heard how much they're moving already, 2 or 3 times today alone. And so we will be using blood-based biomarkers to help us as an inclusion criteria, and readout will be linked with that. We'll be working with the Dementia Trials Network that Cath's already mentioned and the TDRC, and we will also work with JDR, and we're being supported by the Dementia Trials Accelerator. So there are a whole host of acronyms that we'll be working with, and half of the job is just trying to keep an eye on what's happening and all these moving parts to make sure something actually happens. And that, as you've heard from all of us, is quite a job in and of itself, I think. So hopefully, like the good scientists and clinicians that you are, you're thinking about all the problems with what I've said and what the difficulties might be. I just sat for 10 minutes earlier this week and I thought I'll just put some of the things that worry me onto a slide. Language. Half of my patients can't speak enough English to answer some of these questionnaires in English. Placebo. Can we make sure we match placebo? How big are these pills going to be if we're giving metformin and atomoxetine in 2 doses? Something that we don't always say enough about explicitly is the heterogeneity of our patient population and the number of different diseases that they have, the different types of Alzheimer's disease they have, and whether a one-size-fits-all type of approach is going to work. And then if we're giving metformin, people with diabetes won't be able to go in that arm. We have to make sure that they'll go in the other arm. How does that affect things? And then when people are finished, will we re-randomize them? When will we re-randomize them? How long do we wait for? These all, when I stay awake at night, worry me, but they are all surmountable. They are slightly arbitrary things that we need to work on, think about, and come up with a solution for. And I'm not being overly touchy-feely when I say I would be very grateful for anybody's contribution to the thinking about this and how we do it, because that's how we make it better, by people criticizing and then us going back and thinking about it a bit more. So I'd like to finish there. So I showed you Charing Cross Hospital on a miserable day for Monday morning. It's now Friday afternoon, and you won't believe this. This is no filter Charing Cross Hospital almost 5 years ago in the sunshine. I'd like to say thank you to all the people who've been involved, all the people whose names and photos I've already shown, and then some of the team that I work with over there. Thank you very, very much to everyone, and happy to take a few questions.
Often NHS sites are approached to recruit participants into studies, and the experts knowing how to do that really well. Can I ask what input you have with NHS sites when you're setting studies up to know that they run efficiently and stand the best chance of recruiting?
So speaking to site leads, and so Vanessa mentioned at the beginning that I'm the national specialty lead for the RDN. So hopefully that helps a bit, but not completely, in trying to work out how we recruit. One of the things that we've discussed a lot about this is not just recruiting from the same old places, but trying to make sure we work with the trial delivery framework as well as the DTN as well, to those sites to begin with, and then move out. One of the things in O'Shaughnessy is trying to make sure that we do things that make things a bit easier for sites as well. So for instance, not necessarily having to involve local pharmacies, for the blood test, not necessarily having to involve the local freezer, etc. So those are the things that we'll try and push and then see how we do.
Okay, great. Thank you again, Paresh.
I'm an academic neurologist. I work in a teaching hospital, in a kind of ivory tower cognitive disorders clinic. But I'm unusual for a neurologist in that I also go to a community memory clinic and work there with the psychiatrists in a very diverse and deprived part of London, where patients currently have no access to trials. And so that's the sort of context for what I'm going to talk about today. So the trials that we're running currently are not representative, and I suspect I don't need to tell you this. I think it's something that we all know is an issue. There was a review in 2021 showing that 95% of all participants in Alzheimer's disease trials had been white, and there was no improvement over time. But also that a lot of trials were not systematically reporting this. So in some circumstances, it was hard to even see what the ethnicity of the participants was. And we're very excited about these new treatments, but as Cath has mentioned, we don't really know how they work in non-white groups because there are so few people in the studies that we just don't have the data. So in the Clarity lecanemab study, 80% of the participants were white, and only 20 black participants ever received lecanemab in the study. And for donanemab in the Trailblazer, it was 90% white, and only 19 black participants have ever been given it. So we can't really start thinking about safety and efficacy in these groups. And there are a few reasons why representative trials are important. And essentially, it's due to an issue of fairness and also due to an issue of scientific benefit. So we know that and we've shown this in East London, the same people that are underrepresented in trials are disproportionately at high risk of dementia. So people that are getting dementia more are not being included in the trials and not benefiting from the treatments. We know that the pharmacokinetics and pharmacodynamics might differ across different ancestry groups, which can affect safety and treatment effect. But actually differences in treatment effect can tell us something scientific about the disease and what's happening, and give us new mechanistic insight. And being in a trial confers benefit in itself, so including people and giving them the opportunity to be part of the aspiration and the hope, we know that leads to people doing better even when they get placebo, and we need to be fair about giving everyone an equal opportunity to that. But we also need to develop treatments that can be used broadly across the population and lead to equitable benefit. And we need to be producing evidence that is relevant to the whole UK population, because otherwise, when the evidence comes in front of the MHRA and NICE, they are concerned about the lack of applicability to the whole UK population. So thinking about why trials are currently unrepresentative, there is a risk that we attribute too much to medical mistrust among certain demographic groups and a lack of willing. But actually the evidence we have about this is that most of the reason for underrepresentation is structural barriers to participation and not a lack of willing. People want to take part in trials, they're just not being given the opportunity to do it. And I'm going to talk about what the sort of structural barriers are that we face. Broadly speaking, they're around eligibility criteria, where we are running the trials, a lack of culturally and linguistically appropriate trial resources, health and scientific literacy of the different populations we want to recruit, and the kind of time and resource constraints that influence participation. So eligibility criteria, I think, is probably the biggest one. So there's a paper about this looking at what proportion of trials have strict eligibility criteria around these different things. So a large majority of trials exclude people with psychiatric illness, cardiovascular and cerebrovascular disease. Most trials require a caregiver to attend with and be a study partner. More than 90% of trials have specific cognitive test score cutoffs, which we know are culturally unfair. And some trials are now using molecular biomarker cutoffs, and we can see emerging evidence that some of these biomarker levels might systematically vary across ethno-racial groups. In particular, tau. There's quite a bit of evidence now showing that tau levels tend to be lower in African-American people. And so if we have a tau cutoff where you have to be above a certain amount to take part, you'll be structurally excluding a proportion of Black people from that study. And we can see that in the trials that we've all been excited about. So in the lecanemab trial, there were 6.5% Black participants at screening and only 2.3% afterwards. So it's very, very clear that our eligibility criteria are a major structural barrier. And I think we need to go back and think very carefully again about why we have these eligibility criteria, because those of you who work in clinic, if you think about these things, how many patients do you see in clinic who are not depressed, not hypertensive, not diabetic, don't have any white dots on their brain scan? It's very, very few. So we are at risk of, as well as excluding people systematically from the trials, we're at risk of treating something that isn't what Alzheimer's disease is in the real world. I think there's a lot of the time we're pushing to try and run trials in a human version of a mouse model of Alzheimer's disease. And perhaps we just need to be a little bit more pragmatic about it. And that would certainly help with representation. Other study characteristics that are important are the burden of participation. And we know, for example, that when people are living in deprivation and they have more complex lives in lots of ways, asking people to come for more frequent visits and always with a study partner is a major issue. So thinking about whether there are things that we can do to reduce the burden of participation and provide support for people, make the study partner less of a stringent requirement and try and provide that support. Composition of the research team is really important, and we know that if you come from a minoritized ethnic group, you are more likely to agree to participate in a study if you are approached by somebody of the same ethnic background as you. And so making sure that we achieve diversity not just in our participants, but in our research teams is really important. And the trial literature that is given to people tends to be very complicated and overwhelming and in English. And this is a real problem. And if people cannot get their heads around what's in the information, it's very difficult for them to participate. So thinking about how we can provide information in other languages, simplify it, and support people to understand it and take the time to go through it with people and make sure that that's not a barrier. And the other thing is thinking about diversity and representation, paying lip service to it, and then deciding on a fairly tokenistic program of community engagement to try and persuade people to take part. And if you have never worked with a community group before and you drive a bus down there one day and say, take part in our study, people will feel like they're being used. So we need to be more than tokenistic, and we need to build relationships and trust over time, which is hard to do. It's not easy, and it takes time and it takes resource, but that's how we need to be doing it. And we also need to ensure that we're not being tokenistic in the way that we demonstrate the diversity that's been achieved. I've certainly seen studies that have failed to recruit a single black participant, but on the study brochure, there's a black person on the cover in the photo. And it's easy to see how that feels to people, like they're being included in a tokenistic way with that kind of approach. So other things that are important, this is slightly recapitulating what Cath's already shown us about some of the variation by geography. There is a problem globally that we are running trials in the global north in developed countries, and of course we're not getting people from low and middle income countries as a result. We don't really have a lot of data about within country geography, but I think we know that in general trial sites tend to be located in more affluent urban centres. And so thinking about how we can start running trials in areas that are not the usual suspects is really important. And as well as the kind of geography, thinking about the type of site is really important. And this is a particular issue for us in the UK because of the way our dementia services have been split between neurology and psychiatry on the whole, some geriatrics. And most of the trials work that's happening is happening in neurology settings. And the recruiting from memory clinics is very difficult because they're not based in acute trusts that have experience running trials, that have appropriate pharmacy support. They're in community settings. And we know that biomarkers there aren't available, so it's hard to confirm eligibility. We know that only 2% of people in the UK are getting a biomarker-supported diagnosis of Alzheimer's disease. And even if we recruit patients there but require them to travel somewhere else that they're unfamiliar with to take part in the study, it's a barrier to people wanting to take part. And I think that if we could improve our ability to run drug trials in community memory clinic settings, there would be clear benefits. For a start, we would be getting far more people because this is where most people are seen and diagnosed, and would be able to harness what used to be the CRM but is now the RDN infrastructure much better. My experience of memory clinic and psychiatry trust R&D departments is they're super keen to take part in trials. They want to be involved in dementia studies, but they don't have the infrastructure to run drug trials. And you can see this in the figures from 2022 about how many people through the NIHR-funded clinical research network were recruited into Alzheimer's disease drug trials as compared to cardiovascular disease and cancer. And the bar that isn't really there on the left is Alzheimer's disease trials, then cardiovascular disease, then cancer. So it's over 100 times fewer than for cancer drug trials. And it's because we're not running the trials where all the patients are being seen and the teams there don't have the skills to do it. And this is a problem for representation because we know that a certain type of person gets himself referred to a tertiary centre to see the neurology department that has a trials unit. And we know that we've shown that if you are from a more deprived area, you're more likely to have an unspecified dementia diagnosis, which means that you then can't access trials. You've been seen in a community memory clinic. They've just said, well, it's dementia, but we don't know how to subtype it. And then you really have an opportunity to take part in this type of research. And I think having community sites allows you to build on existing trust and community links. Most memory clinics are quite well linked in with some of their local community organisations and have a working relationship and have trust that you can build on that would then be a different type of engagement to somebody driving a bus there one day. And I think that memory clinics are also perhaps more likely to have staffing that is representative of people living in the local area. So here are the things that I think we should be doing differently. So that I think we should be improving our reporting of representation and really viewing it as a priority. And I think we should be producing, figures at a national level with the kind of RDN figures about who's been recruited from which demographic group. I think that we need to consider population comorbidities and social determinants very carefully when defining study entry criteria, and avoiding strict cutoffs on the MMSE, for example, because we know it's not culturally fair. If we need to include people on the basis of a certain level of severity, we could perhaps think about activities of daily living which are more culturally fair than pen and paper cognitive tests. We need to try to think carefully about biomarker cutoffs and how they're excluding people structurally, whether we need to make some adjustments, and we need more evidence to support that. And that's the sort of evidence that we will hopefully be getting from studies like Readout about variation in biomarker levels. We need sites in diverse areas and community settings where we need to kind of optimize the facilities and the staffing for screening activities to make sure that those things can happen in busy memory clinics that are very stretched so that they can still take part. We need to provide better support for participants and care partners to reduce barriers. And we need to produce our study documents in other languages in a simplified way and support people with going through them and understanding them. And we need to build on the relationships that we've got with community groups. It's very difficult for a pharma company running a study to develop those relationships with groups all around the country, but as researchers, we can in our local area. So the bit that this is rolled back to a previous version when I sent it, unfortunately, but what I was hoping to show at the end was some of the work that we've done with local groups in our area. So we work with the Hackney Caribbean Elderly Organisation in East London. We've been developing a partnership with them over the last 5 or 6 years. We go back there year after year, study after study, to consult with them, to have them feed into our research. We've produced new research based on things that they've told us. So the director of the Hackney Caribbean Elderly Organisation told me that all her members with dementia were telling her about aches and pains and physical symptoms they were having, and none of them were talking about memory, and shouldn't we look into this? And so we've done exactly that. We've just had a paper come out showing that if you're from a non-white ethnic group, you're more likely to have non-cognitive symptoms of dementia presenting to primary care, and this could be a way of improving equitable diagnosis. So we're now going back to them as a group and saying, we heard what you said, we've done some research to look at exactly that issue, it's now published, we've included members of the group as co-authors in our work, and that's the type of relationship that if we were now to go to them and say, we're now running a drug trial, would you like to help us persuade your members to take part? You could really build on a trusting relationship. And that's the sort of thing that we need to be developing. And as researchers, we need to be developing it locally for ourselves in our area so that we have a network of those sorts of relationships around the country to build on. And a part of that is really demonstrating that we are committed to these groups beyond the trial. Beforehand in building the relationship, and then afterwards in coming back again in sharing the results and in asking people to input into the design of the next study that we do. Okay, I'll stop there. I'll take any questions.
One thing that struck me when you were talking about diversity and communication was I've been involved, I know like others recently, in studies with people with Down syndrome, obviously massively at risk of Alzheimer's disease, and we're starting to see commercial trials being done in that group. And I've certainly been involved as a site trying to recruit people with Down syndrome and recognizing this is a whole other group in terms of an underrepresented population. And actually, they're not largely sitting in secondary care, but a whole other level of outreach and a population, as I said, that we're not frankly used to supporting and working with. And I wonder whether you think how many of your experiences in dealing with a variety of groups locally to you and some of the stuff you've just mentioned that you found has made a real difference for the populations and recruitment could be applicable to other types of underrepresented groups and trials.
Yeah, I think so. And I think a major issue for people with Down syndrome is that they don't tend to end up in the memory clinic because they're seen in LD services. So it's something that we are kind of completely cut off from. And yes, I think we need to, I've spoken a lot about kind of ethno-racial diversity, but yes, lots of other things that we need to think about, including people with Down syndrome and how to bring them in.
That's great talk. I agree with everything you mentioned, really, about trying to improve equitable trial delivery. I think there are two things I just wanted to add. I think, firstly, you've sort of glossed over cultural barriers to ethnic minority representation. And I think that they are there, and I think we need to address them. So from my own personal experience of looking at the clinical phenotype of African, Caribbean and Indian Parkinsonism in London, I found it more difficult to recruit patients unless I knew them and I'd actually looked after them in clinic, for example, and made a personal relationship. Some were Jehovah's Witnesses and were not happy to have a PET scan, for example, with injection of radioactive dye. And then also the other thing to think about is actually going out into sub-Saharan Africa. And I just, as part of that, and looking at the phenotype that's there and comparing that, say, to second or third generation people living in the UK. And it's just a plug for an NIHR-funded grant called TRAP-CAF, which I'm involved in, which stands for Transforming Parkinson's Care Across Africa, which is involving 7 countries across Africa and is phenotyping and doing exactly that. And as a result, the community in Lagos, Nigeria, have identified a novel GBA gene variant that's never been described before in Parkinson's. So.
Yeah, I think that last bit speaks to the scientific benefit of doing this over and above the fairness benefit. I think so my experience has been that whilst there are cultural barriers and some of those are related to stigma, that actually if you engage and get people talking about these things, and if you specifically tell people that there is a problem with fairness and representation in research that you can help us to address, then you do overcome quite a few of those barriers. And my last slide was going to be Lullin, who's the director of the Hackney Caribbean Elderly Organisation, on a video saying that she feels that her job is to break down the stigma and raise awareness in her community so that she can then get people talking and bring them in to take part in research so that they're represented. So.
Okay, nothing else at the moment, so thanks again.
Thank you.
Good afternoon, everyone. And firstly, thank you so much for the invitation to come here today. It's a real pleasure. And I'm really humbled and honoured to be in your company. And like most of you, I am not a neurologist. I'm actually a trained parasitologist. So that's an interesting route from parasitology straight into dementia. So obviously lots of stories to be told on that one someday. So, so I'm just going to really talk a little bit about what we've been thinking about in terms of Roche for advancing our clinical trials across the UK. So, obviously, there is a real multitude of initiatives across the UK, and we're really, really privileged to have this rich ecosystem of research across the nation. However, it has to be said, if we're being totally honest, it is quite complex to navigate all of these different groups. And each of them have slightly different nuances about what their objectives are. So what has been a real pleasure to see over the past few months is really the initiatives as the dementia programme's goals, and also seeing the coalition of NIHR and DPUK and the DTN sort of have this much more position of having one shop window for industry, because what is really important for us is to really be able to navigate the system and be able to step through the door at the right time for the right objective for the right project. And when actually we do step through the door and we sit together, it's really obvious that we have totally synergistic goals and that map directly with industry vision and the network's vision. And I'm not going to go through all of these things on the slide, it's a busy slide, but really just to recognise that the how in what we're trying to deliver, so centralised imaging qualification, centralised feasibility, having joint educational materials, these are all things that together we can really make an impact on. I'm not going to talk about them all, but I'm going to highlight a few of these things that we've been talking about and focusing on. So one of them is race training. I think this is probably a recognised bottleneck across all of our trials trying to find qualified raters. So, and I think it has to be said that as industry, we don't do a very good job. We set a bar of what qualification needs to be. We don't really let you know what that bar looks like. And then when you step up and say, we've got a rater here that is we'd like to put forward for your trial, you get a binary, yes, you're qualifiable, or no, sorry, you're not. So I think this is an area that we've really wanted to improve upon. And one of the things that we've been looking at is working really closely with the NIHR Rater Foundational Training Programme. And this is really a fantastic piece of work, and really want to applaud this programme. And what we've done is really try to set out and look at pathways of getting people from the foundational level and the NIHR programme into our industry trials, and looking at that sometimes there are different levels of experience, and therefore, you know, if you are a developing rater, you might need more practice, and giving those pathways and making them a lot more transparent. We ran a little pilot with 4 sites, and we learnt a lot. It was a really great learning experience. But I have to say, we did have a bit of an aha moment, in that it was recognised that the institutes that we were working with were the real, large, hugely, I think somebody called them the ivory towers earlier, but the well-loved institutes. So we really started to think about, well, okay, that's one objective where there's lots of infrastructure, but what about broadening infrastructure in areas where we don't have as much infrastructure, in the more rural communities? And that's where, if we start thinking about other ideas, like, is there opportunities to train a more mobile or roaming raters or agile workforce that could really help manage the ebbs and flows? And I think it was also recognised earlier about this inter-rater variability. So if there is a pool of trained raters that can support sites within regions that can go out and manage these resourcing gaps, then I think that's a real win-win. And the other thing, that if we think about this model one step further and we say, okay, could we actually build a bilingual pool of roaming raters, and actually then how we could utilise those across the communities? You know, with sponsors, when we're running global trials, we often validate our scales in 20 languages for a big global phase 3. So we have those scales licensed and validated in our trials. So I would suggest that there could be other ways that we could use them in our communities across the UK. So this is a point that I really wanted to agree with Charles in reference to what he was indicating with community initiatives and going out into the community. Because there are some great initiatives and infrastructure that we can leverage to go out into the community to talk about clinical trial research and even do early identification. However, I think what we have to do is really use these services with intentionality. When we step into the communities, we really have to think about, and then what? And then what for people living with Alzheimer's? Often these community engagement activities can be the first introduction to clinical research for these people. So we really want to maximise their experience. These individuals are going to be our advocates for research within their communities. So we really want to make sure that they're having the best experience. We don't wanting to step into these communities, pick them up and drop them. We really need to think about them end to end in their treatment and diagnosis journey. What are we giving back? How are we going to support them and signpost them to the right places? And what language are we using? So are we using the term pre-screening, which is a term that we love in industry, but actually, how does that resonate in the community? Should we be thinking about terminology like self-assessment or risk assessment? And then providing clarity. What's next for me? When will I get my results? How will this be communicated? How will I understand my risk? Making these these terms more culturally appropriate. And then, as I said, making sure that these individuals are then signposted to either support groups, advocacy, or that they're receiving the right levels of education. Community-based screening can enable sites to be more flexible. They can support outreach and engagement. But if we look at this model, maybe we can be even more effective with examples where we consider trial agnostic screening. So really, instead of having these large screen failure rates of clinical trials, maybe there's an opportunity to say, thank you for participating. We are now going to signpost you to the correct trial at the correct time. And this obviously then gives us the opportunity to also build dialogue and further trust and build sustainability with these communities. Other opportunities that obviously we'd like to talk about, and this is a great one, is the Readout Project, and obviously led by Vanessa's group with the DPUK, and how we are looking at being able to flag individuals that come into these sort of observational projects into interventional trials. But they're not thinking about them just one directionally. We need to think about these bi-directionally. So if people aren't suitable for, or have been even in an interventional screening process, but have been told they're not suitable for trial X, can they actually then go back into a registry so that they can be followed up and then maybe offered future opportunities? One example I did want to share with you, and because this was a real learning for us, in one of our prevention trials we ran a mobile research unit through the collaboration of GAP, and we called this mobile research Thor, which was taking healthcare on the road. And what we did within a very short time period is there was 20 community events run. Within those 20 events, we managed to identify and pre-screen 325 individuals, and just over 50% of those were from diverse and underrepresented communities. So we also thought that we needed to follow up with the staff and really understand what had worked best for them. And what we heard was that the greatest opportunity here was that they were enabled by the bus to go out into the community, but it was the bus was staffed with the actual site team that were bilingual and were representative of their local communities. So when they went and visited these individuals and talked about brain health and also took a blood-based biomarker sample, they were able to maintain the continuum of care. The same people that they saw in their community was the same person that then picked up the phone and was the same person that then saw them in clinic. And what they said when we surveyed them was 80% of the staff agreed that Thor had a positive impact on building relationships. And actually what Thor also demonstrated is that they could increase pre-screening in the community by 126%. So one of the things that we put a lot of value in is understanding trial experiences. We really want to understand what matters most to maximise people's experience and to maximise inclusivity in our trials. We globally have engaged with a number of patient advocacy groups and released a couple of publications, and we've got some co-authors in the room and sitting at the back, so shout out over there. Also want to give a real demonstrate how interested we are in sort of the portals and POPT that I know Vanessa referenced earlier, but also one of the initiatives from Cath's group was around badging sites from her network that are really focused on trial experience and participation. And that's really helpful for us, because if there is a site that is we can recognise that puts a lot of resource and focus in this, they can be obviously flagged to us for sort of site feasibility and such like. However, I did just want to share a couple of slides with you, because I think that if we want to really talk about making an impact and actually thinking about how we have to broaden our educational efforts and what the scope of that, and to understand the lift of what that could look like. I was looking at, first of all, the World Alzheimer's Report. And interestingly, and I found this fascinating, that in 2019, this report said that 62% of HPCs believed that dementia was a normal part of aging. They ran this survey again in 2024, 5 years later, and this had gone up actually to 65%. However, over 90% of carers and respondents from the public stated that if they knew that there was a medicine that could prevent the progression of dementia, it would encourage them to get a diagnosis. So this led me to be curious about what our stats were in the UK. So I looked at Alzheimer's UK report and the Dementia Attitudes report from 2023. So showing actually that the UK actually thinks slightly differently. Actually, it was quite nice to see that 65% of people disagree that dementia is an inevitable part of aging. But similarly, almost 90% of people would seek a diagnosis. So I guess the point I'm trying to make here is that we need to improve the dialogue. We need to make it more common, have common language that we can all talk to on every level, so that we can offer clinical trials not just to the few, but to the much broader generalizable population. Let's move now. So just to wrap up, and really just to identify that we are really working to try and close the gap. And we do have a very unique environment across the UK with scalable opportunities, but resources are not infinite. So I pose a point for us to ponder on. If there was an opportunity for us to co-create a UK playbook that identified clear access points between industry and the UK resources, that we could really demonstrate best practices, highlight how to navigate some of the systems, I'm sure that we could come up with a really good demonstration of how we can advance inclusive trials across the UK. We really want to be able to make our trials sustainable. We want to maximise and leverage the efficiencies of resources. And I think that, as Cath alluded to earlier, we want to really put the UK at the global stage and really leading the way for advancing AD clinical trials. So, thank you very much.
Thanks very much, Ruth. And I think the statistics you showed were really striking, weren't they? As you said, more positively from AR UK, but slightly concerning that healthcare professionals still seem somewhat misinformed about possibilities. So there's still a huge amount to do. And I completely agree. I mean, I know as a clinician that does research, and I know we've talked a lot about this, that I still think there's a huge conversation we need to have with the public about what's happening, what's likely to happen next, and what they actually want for services, and around biomarker characterization, diagnosis, etc. Because I think we're still dealing with a lot of ignorance, unfortunately, where people are just not aware of a lot of what's going on. But any questions for Ruth at this point?
Hi, Ruth. Thank you for your interesting talk. I was interested in the fact that you gave about 80% of people would want to know their risk if they thought a treatment was available. So my research is looking more at sort of modifiable factors that we can influence, so like lifestyle factors, exercise, diet, that we can change. Have you any experience of what people think about that? Because we know it doesn't seem to work with risk for obesity, risk for heart disease. But we do know that we can influence the outcome, or at least the onset of some of these conditions, by leading healthy lifestyles. Have you any experience of what potential patients might think about that?
Yeah, so I mean, some of the work that we did with GAP actually was that they do some great community engagement activities. That they have an Activate Brain program. And that's a really nice initiative where they go out into the communities and talk about lifestyle factors and risk factors, and really sort of talk to individuals about their brain health. And that's really, really well received. I can definitely. The data the greater than 90%. That's it's not my metric, I'm afraid. But I totally agree that from what I have seen when utilising those sort of messages, it's a great way to introduce. The way that I was discussing it with someone is that you start with a funnel. You go out more broadly and you start talking about brain health, and then you sort of can narrow the conversation down to sort of more clinical trials. But you can't just go in and go, bang, let's talk about clinical trials. You need to sort of go in and sort of talk more generally what people can do about their lifestyle, decrease their risk, and then, bring the conversation in towards clinical trials.
Thanks. And I'm guessing, Ruth, probably a lot of people are very receptive to those conversations. But I guess, it's that lack of education, of awareness that there's things people can do in terms of lifestyle changes that will have an impact. Any other questions for Ruth?
Thanks very much. I just wanted to ask about difference in commercial and NHS trial sites, and what we can learn as NHS sites from the commercial sites, which seem to be where most trials are happening, late phase trials are happening in Alzheimer's disease.
Sorry, just to clarify.
So I guess the Roche trial that's running at the moment, the brain shuttle trial, I think 3 of the 4 sites in the UK are commercial, run by commercial organisations. My experience trying to get trials set up is obviously that's much more attractive to companies who want to run a trial. They seem to be maybe faster, more efficient. I'm just wondering what we can learn as clinical NHS sites to improve how attractive we are for clinical trials.
So I think it's everything that we've been talking about, and I think it's the exact aims of DPUK and the DTN about how we can. And I'll give you one of my favourite stories recently of one of our interventional trials in the UK. They were setting up sites, and commercial and NHS, and using the model of having a pre-qualified central imaging site, using the agreed NIHR costing template, and using pre-qualified raters through the Rater Academy I was highlighting. We managed to get an NHS site from site selection to activation in 36 working days, and that site beat the commercial sites. So it can be done. It can absolutely be done. I think we just have to be really intentional about the activities where we put our efforts.
I'm Sasha. I'm a postdoc from the US of Manchester in the Cooper Lab Group. And today I'm going to talk about the use of sodium as a possible biomarker for vascular dementia. So vascular dementia is the second most common form of dementia in the elderly, mainly caused by lack of blood flow to certain areas in the brain. And this right here, can't really see it, is a very common, or very simple schematic of some of the more common pathologies you might see in someone with vascular dementia. So you have bleeding of different sizes, you have infarction, and you have sometimes, in some cases, cerebral angiopathy, which can also apparent in other dementias as well. And this can lead to cognitive impairments, it can lead to speech difficulties, behavioural abnormalities, and lastly, motor dysfunction. But the main thing here is that everything you can see on this slide here can also happen in someone who has Alzheimer's disease. And this makes it really hard to clinically diagnose vascular dementia. And so we need better biomarkers for vascular dementia to improve patient care and to select new treatments. And I think this is where my research can come into play. So what I do and what my lab does is it's a very mass spec-based lab. So for those who don't know what this is a way of analysing a molecule by measuring its mass-to-charge ratio. I won't go into too much depth, but I just want to explore the fundamentals of mass spec before I go ahead. So every mass spec will have 3 main components. This is the mass spec here at the top. You'll have an ion source, a mass analyzer, and a detector. So your sample will go through this mass spec, and you'll get what's known as a mass spectrum at the bottom here. This is essentially a unique fingerprint for that molecule. And so you can analyze that molecule to analyze that fingerprint to understand what molecule that is and how much of that molecule is present within your sample. So extremely sensitive, extremely robust. And there are loads of applications for mass spectrometry. One of those applications is looking at metals in various tissues. And this can be done using this approach here, using ICP-MS. And so we actually know quite a lot about a variety of different metals in various diseases, and diseases like Alzheimer's disease, frontotemporal dementia, Lewy body dementia. But by comparison, we know very little about these metals in vascular dementia. So that was my kind of rationale for doing this project. So I kind of set out to rectify this by doing a multi-regional analysis, looking at 7 regions in cases with vascular dementia and matched controls. So I put these regions through this ICP-MS, this mass spec, and I saw quite a striking phenotype. So as you can expect, I saw huge elevation of sodium throughout the brains in people with vascular dementia. And this was a multi-regional phenotype, so it isn't restricted just to one region. And this increase was around 25% higher in VAD compared to controls. So a really striking phenotype. And so I put this kind of data onto this schematic to make it a bit easier to understand. So same as for the previous slide, areas in red represent areas of increased sodium in the brain, areas in purple represent no change. And interestingly, my group also has corresponding post-mortem sodium data from people who die with Alzheimer's disease using the same techniques at ICP-MS. And so I wanted to compare these two dimensions, these two diseases. And this is what I found. So straight away, you can see that there are now 2 different or contrasting patterns of sodium in the brains at post-mortem. On one hand, in vascular dementia, it's more widespread, as you can tell from these kind of red regions around the brain, whereas Alzheimer's disease is more localized to the middle of the brain, this kind of around the middle temporal gyrus. And we know that these regions are severely affected in Alzheimer's disease at post-mortem. So you may be thinking, okay, great, now we have this biomarker, or this phenotype in these 2 dimensions at post-mortem. How can this be used as a biomarker? Well, fortunately for me, we can actually image sodium in real time in patients. And this can be done using sodium MRI. This is a figure taken from one of my collaborators, Dr. Niels Muellert at University of Manchester. So this is essentially a quantitative sodium MR image in a healthy control. And if you see these kind of yellow regions here, this is elevation of sodium within the brain in real time. So we can actually do this in healthy controls. It's been done before in patients with multiple sclerosis, in patients with various brain cancers, but it hasn't yet been applied to vascular dementia or VCI. So going forward, I'll be using this technique to try and more accurately diagnose people with vascular cognitive impairments and see if we can establish a better biomarker for this disease. So the next year or so I should have some really interesting research, really data. So definitely watch this space. So, yeah, that's it. So thanks to everyone in my lab, everyone else who helped with the work, Southwest Dementia Brain Bank for supplying the tissue, and lastly, my funders, the Lee Research Trust. Thank you for listening. Any questions?
Do you have a hypothesis as to why you think that might be the case?
Well, for the widespread sodium?
Yeah.
Yeah. So this has been heavily researched in multiple sclerosis, and I think I agree with what they said. So they think it's to do with the sodium potassium pump. It's just not working due to lack of ATP bioavailability. And so less ATP leads to reduced activity of this pump. So you're essentially getting an increase in sodium within the cell.
Great talk. So I was wondering if you have any other imaging data from these same individuals. So are you seeing these sodium changes in regions that are hypoperfused or that have microinfarcts, for example?
So I haven't started the imaging at all. I will in the next year or so. So I'm currently recruiting. But yeah, we will be doing T1 weight MR, TT flare to look at lesioning, white matter lesioning, as well as ASL as well to look at the blood flow in the brain. So yeah, we will have that data very soon. But yeah.
Thank you.
Thank you. It is very interesting that the sodium pattern in the vascular dementia is localized because the vascular lesions are maybe focal, usually focal. So what do you think the reason for that finding?
I'm not too sure, actually. I thought, same as you, it would be kind of more localized to, say, the basal ganglia and the thalamus, which are more affected in vascular dementia. The fact that we're seeing it's widespread is an interesting finding, which is why I want to kind of probably a bit more using kind of clinical imaging techniques, but I'm not too sure why it's widespread.
Okay, we'll finish there. Thank you again, Dr. Philbert.
Hi, everyone. My name is Mats. I'm a postdoctoral researcher at the Oxford Centre for Human Brain Activity, where I work with Professor Mark Woolrich. There we go. And I would like to give you a recent update of some findings from the New Therapeutics in Alzheimer's Disease study, or NTAD. So this is a multi-center study led by Professor James Rowe, where we aim to find Alzheimer's disease biomarkers based on electrophysiology. And particularly, I will be looking at magnetoencephalography, or MEG, which measures postsynaptic currents in populations of neurons. The most characteristic metric of this type of brain activity is the spectral profile of the data. And as we know that amyloid beta and tau protein aggregates in the brain, they directly affect synaptic transmission. And because MEG directly measures these postsynaptic potentials, it has the potential to also pick up on on synaptic changes due to protein aggregates. So a bit more about the NTAD study. In NTAD, patient status was based on the presence of amyloid beta based on lumbar puncture or PET scan. And patients and controls came to the sites for 3 times. So there were 3 visits. There was a baseline visit in which 65 patients and 35 controls came to the institute. And we can use this data to look at the cross-sectional differences. Then 20 patients came back after 2 weeks, and that data can be used to look at retest reliability. And then the annual data, the annual visit, we had about 45 patients come back 1 year later, and that data can be used to look at disease progression. So we have quite a complete data set in that sense. As I said, the most characteristic metric to look at this type of brain activity is the spectral activity. And that has been done quite a bit in the literature. And what people have found, we replicated initially. So people tend to see this decrease or like a slowing in the activity. So there's there's an increase in low frequency power, so delta and theta activity in patients, and a decrease in beta activity. So that's nice that we can replicate this. But one of the caveats is that the effect sizes are very small. So they reach only about 0.2 in terms of effect size. So it's quite unlikely that this is going to be very informative on the individual subject basis. And also, when we look at the reliability of these metrics, they're only moderately reliable. So again, that's not super promising. And then looking at disease progression, we actually didn't find any differences between the baseline and the annual scan. But one of the big assumptions in this analysis is that the activity is static over time, and we know that that is a very crude assumption that is not true. So instead what we did is we adopted a network perspective where we assumed that at each moment in time, a specific brain network is active, and each network has their own characteristic spectral profile. And when we do that and we repeat these analyses, we find that cross-sectionally, there's a similar pattern of a slowing of activity that's present in almost all states. But different states are sensitive to different parts of these changes. So some states are sensitive to the delta theta band effect, and some states are sensitive to the beta effect. But importantly, the effect sizes are much stronger. So here we get up to a strong effect size of 0.6 in the beta band activity for a parietal alpha state. They're also much more reliable metrics. With the exception of state 1, which is the default mode network, all states had very good test-retest reliability. And even we even found some sensitivity to disease progression. So when we looked at the differences between baseline and annual scans for patients, there was one state, which is a state with a lot of frontal activity in the frontal lobe, that showed differences in the high gamma band between 80 and 120 hertz. Again, with quite strong effect size. It's quite interesting to see that the potential biomarker that we're looking at here is different for cross-sectional differences and for disease progression as well. So that's what I'm looking into now. I'm seeing like how well can these metrics be used for prediction at the individual subject level. I still have my poster up in case you want to know more details, so please come and see me after the talk. Thanks.
Thank you for your talk. Can I ask a question of how you derived your components? Were they sort of independent component sort of analyses? And secondly, can I sort of ask whether, actually, I think I've forgotten. Yeah, I mean, yeah, can you tell me what the components first? I think I've forgotten the second question.
So these states, they were determined based on a hidden Markov model, which is a quite well-established approach to infer these kind of resting state networks from MEG data. And these are quite prototypical networks that we see time and again when we use this method on different data sets. And they resemble quite well the networks that we know from the fMRI literature.
And secondly, can I ask, was this all sort of resting state data? And if so, have you looked at task-based?
Yeah, I should have mentioned that. So here I've been only looking at the resting state data, eyes closed. We do have task data, that we have 4 different tasks. I started looking at resting state data mostly because it would be ideal for patients to come in and not having to do anything, because sometimes these tasks that we use can be quite strenuous.
And how long was the resting state?
Only 5 minutes.
5 minutes, okay.
Yeah.
Thank you.
There were quite a few very interesting talks about therapies. I would like to know your opinion about your perfect way of measuring response to any therapy. And with existing or not yet existing biomarkers, so if it's something existent, that's one thing, or what do you think would be the best biomarker or the way of measuring that we're doing something really good? Because, you know, if cancer, it's relatively, I would say, easier because we can see the tumor is disappearing. How do we see that in dementia? What's the most robust way from your perspective of measuring that? Thank you.
Yeah, please feel free.
First of all, I've just realized in my talk, I didn't thank Vanessa as co-lead of AD Smart because you were chairing. So I have to do that first. I've just remembered. Then the second thing is, if it's all right to answer, I think that the best biomarker is the person who is affected being able to do the things they normally do. So it sounds like an old man's answer, and I guess I have become an old man, but in clinic, that's what we rely on most. And I think everything else is trying to get to that. And even that's how we tell people have got worse. And I think that's how we tell people have, when we're succeeded.
I suppose if I can interject, though, I guess one of the things, Pavel, you may be getting at is what if people are not doing anything differently at that particular stage? How are we going to ascertain change?
I'm not sure we need to. So I think that's what I'm trying to get at, that it's the actual ability to do things is what we're sort of asymptoting, whatever we're trying to do. And then whoever brought it up about amyloidosis or not at any stage, what we're trying to do is make sure nothing happens to what people can do at home. So when we're thinking about trial biomarkers, passive ways of getting at that, I think is very important.
Yep. Any other thoughts from the panel?
Yeah, I mean, I think the really glaring thing that we lack as a field is some really good, robust and dynamic biomarkers of brain network function, which is why we get excited about MEG that we just heard about, because I think that's the thing that we're missing. We're getting really good at measuring the presence of toxic proteins, But in between that and the functional outcomes that are so important, we had some better endophenotypes for when the brain function was changing. That could help as a proximity marker for those people who've got a brain full of amyloid, and we'd want to know whether they're the ones that are going to develop functional impairment in the next 5 years to go into a trial, but also in kind of dynamically measuring response in the short term in those trials. It could be a great way of throwing people out of smart AD because something isn't working if you had something that's dynamic enough to get that short-term response in terms of brain function.
Ruth, I'm going to put you on the spot and say, do you have an industry comment on this?
Ooh, I think I need my, but we've got my esteemed colleagues from our biomarker group. I mean, obviously, there are lots of interest on, I think, moving towards digital biomarkers and that sort of space. I think having these validated is really, really helpful. I think we need more data, perhaps, to be able to put them in our pivotal trials, but I think we should, it's really interesting to look at ways of how we can integrate them better earlier on into our trials. And certainly if we are considering going earlier in the disease and into, you know, pre-symptomatic, secondary prevention, even primary prevention, you're going to need sort of tools that can operate at a scalable level, and that's a level that doesn't include certainly PET or CSF.
Just to add to that, I mean, it's a classic question I think we all ask all the time. It very much depends on the stage of trial you're in, and it depends on the stage of disease you're in, and to the point about amyloidosis and how do you tell if somebody hasn't got any symptoms. Looking at different trial designs where you're looking at symptoms not starting, you're looking at events, time to events, rather than function. But I completely agree with what you've heard, that the bottom line is that these treatments, you use your mechanistic biomarkers to have your quick yes, no, it works, it doesn't work. You use your functional biomarkers, and we haven't got good enough ones yet, completely agree, to show you that you're making a difference to the individual. And a difference to the individual is another thing that we can't tell yet, because everything we have is a biomarker that's used in a huge population on average. So there's a lot of improvement we can make in what we need to measure. And so we can't really answer your question at the minute. We're just going to have to keep working on it.
Good point. Other questions, please, for the panel. Tim.
Thanks. I've got two. Well, I'll go for the controversial question. Can I make an argument, and I'm not sure how much I agree with this, that we don't know enough about the processes in dementia yet to even start trials? As my controversial statement. If you look, you could argue, looking at the studies we have, we've got rid of amyloid, it's made very little difference. Is amyloid relevant to the Alzheimer's disease? We don't know. In inflammation, some inflammation seems to be protective, some seems to make things worse. Don't we need to know more about the mechanisms before we start trials?
Who wants to take that first?
I'm going to say something very brief and blunt, Tim. You've been talking to the wrong people. But apart from that, if we waited till we knew everything, we'd never get anywhere. We have to start with what we know and build on it. We do know that there is a very good genetic basis for the hypothesis that we have that amyloid is important. We're evolving that all the time in terms of what it really means. You ask John Hardy now, he'll tell you a very different story to what he would say in 1984. And so as we learn, we develop. What have we learned from the past 20 years? A huge amount, which is why we've got the beginning that we've got right now, which is no small step. So yeah, no, we don't wait till we know everything. We start now and build on it.
I would just say that trials are a great way of learning more about mechanisms as well.
Exactly.
Tim, did you say you had a second point after your controversial statement?
Well, second, slightly less controversial. The problem, the challenge of that we have in clinic is that most people come to see us when they're quite advanced. How do we pick up people earlier on when they're most likely to benefit from therapies? It's not unusual for us to get people who've got an ASA of 50 or 60 at the time of diagnosis.
So there are plenty of initiatives and accelerators and things like that trying to do exactly what you've just said. I think at the moment our system, I'm just looking around to see if I'm going to get into trouble. Our system is not entirely fit for that purpose. The way that people are referred and then seen and then wait, and we heard about that already today. So I think looking at risk and trying to enrich the population that we see according to their risk, and then also being a little less siloed. So neurologists seeing people with cognitive disorders, other people seeing people in a memory clinic, and other people seeing in care of the elderly / geriatrics clinics causes problems, because we know lots of people start off, you published on this the other day, with neuropsychiatric symptoms, and then they bounce from clinic to clinic before being seen months, if not years later. So I think having a bit more open mind about people who are starting to have subjective problems, and a much more streamlined service, which is not very easy.
Yes, so I'm not going to make an NHS England comment based on that.
It doesn't exist anymore. Two things. Firstly, reframe. Why are we talking about dementia? Why aren't we talking about brain health? If we were talking about brain health, then we'd have a far healthier population and probably a far lower population of people with dementia. If we look at risk, what's good for your heart, what's good for your health, and we're doing that in well people clinics, brain health clinics, that's the way to identify people and start with prevention, not early enough, but at least better than we're doing now. And we will identify people, and already are identifying people much earlier. And the second is that just an observation from doing studies with people that are asymptomatic but amyloid positive. It's very interesting how many people want to know, want to get, I won't do the round the table thing here, but if you do that in an audience, you tend to get around 60% of people at least want to know and want to see their amyloid scan, etc., as long as you've got robust processes in place. So actually the public, as Ruth was saying, public want to diagnose this and want to be aware. It's the structural issues that we have in the way at the minute, like we need to reframe, that we need to get better at.
I would just add that I think the only way to deliver that kind of shift is to have treatments available, because some of it is a product of nihilism. So I think we need the treatments to make it happen.
But that's already making a difference. Even though we can't give these treatments right now, we have people coming much earlier or getting their own ApoE4 testing, all the rest of those things, which is actually a really difficult position to be in. But it suggests that that is there, isn't it, that drive?
I have a question, sorry. In the last 10 years, from my understanding, we have made a lot of progress in the way we diagnose and the way we manage patients. What would the panel think in the last 10 years? What was the key finding, or which would be the one item which you would think has made a big difference in the identification or disease process, or in terms of management? Also, second question related to that, in the next 10 years, what would you predict will be the key thing? If this was located or this was found, we would have gone far ahead in the treatment. Thank you.
Paresh, you're looking at me, that maybe you have an answer for the next 10 years.
I can do next 10 years, I think, speaks to what Donna was talking about yesterday. And I think the blood biomarkers, as they come through for the different pathologies, will mean that the words we use for these conditions become out of date. And we will not be saying Alzheimer's disease. We will not be saying Parkinson's disease. We're saying this person has, amyloid, tau, alpha-synuclein. Yeah. These are the three things that are involved, and we will approach it on a much more, dare I say it, scientific basis. I'll leave the last 10 years for someone else.
Okay, and/or the other question was the biggest achievement really in the last 10 years that's changed things.
Sort of not quite. I think what's been remarkable over the last 10 years is, despite all the development, so little of it has made it into general clinical practice. So, we've heard about state-of-the-art approaches to brain image analysis, but we live in a country where more than 50% of people never have a brain scan on the way to getting a dementia diagnosis. So we seem to have found ourselves with a system that actively resists these amazing developments. So I think we need to think about how to address that. And I think some of it relates to nihilism about not being able to do anything. Some of it relates to ageism, frankly, about, we're still, even as professionals, very tied up in these things being diseases of aging, that we don't give parity with other diseases like cancer. So my hope for the next 10 years is we actually start seeing these things come into clinical practice and start seeing memory clinics demanding, I want my patients to have an MRI scan, I want my patients to have biomarkers, I want my patients to have access to research, and less of the attitude that I still encounter, which is, I don't need a brain scan to diagnose dementia, I can do it with an MMSE, what's the point? You know, you still come across that a lot. So it's not quite an answer.
And I'd kind of add to that, I remember still very clearly a patient's relative saying to me, well, why do we need to come and see you in clinic? Can't you diagnose dementia with a questionnaire? So, which was a valid question, I think, if people, again, are not educated in terms of this is not normal aging, and there's still a lot of that sense out there. Other questions for the panel? Over here, Simon.
Fantastic information for all of us to keep on board as we go forward with our research. I have two parts to my question. So given the wealth of knowledge that we now have and we are learning about, and we will be learning about, how can we make what we learn more digestible for the patient that you see in a clinic? Because for most of them, they will come into a clinic and they'll walk in and then they'll see all these fancy things that we've developed. But for them, they just may want to know yes or no. You know, what can we do to really help give what we know more translatable to patients? And then secondly, this is more in relation to the sort of drugs that are coming onto the market. How can we manage the expectations of the general public who have such high hopes for us researchers to have a cure? Because we always talk about these great developments, you know, in the news, lecanemab, donanemab, trontinemab. But these people want to see tangible results, and how can we really manage that so that we don't, let's say, overblow their expectations?
Really, really important. We know that between 10 and 15% of people are going to be eligible for these treatments, at least initially, if we ever manage to give them in this country. That everything you've asked is about communication, isn't it? And I think that the advent of these therapies is going to dramatically change the way that we communicate with our patients. It's going to become a shared decision-making process. It is to some extent right now, but we don't have to take into account so many other details. They don't have to take into account so many other details. So we're going to need to develop tools so that we can understand somebody's values, somebody's priorities, somebody's understanding, their methods of deciding whether something resonates with them or not, because it's going to be different for every person. And we're going to need to have an understanding ourselves that this is a discussion. This is not a giving information. This is about a two-way process of helping them make a decision with their family, their care partner, etc. There are very various ways of doing that, but we're just beginning to learn them now. And it comes back to that point about globalization and international working. Understanding what's happening right now in the States, what's happening in Japan, what's happening in Korea, how they are changing those things. And Donna's here, who gave a talk recently about the heavy lifting you have to do when you start to put these, not just the structural, but also the soft processes in place, right, to make sure it works. It's not easy. And we suffer with a lack of resource in our memory clinics, as well as primary care and everywhere else, in terms of how to do that. But that's what we're going to have to do. So I think it's learning from the people that are doing it already and understanding that it really is going to require quite a lot of work to get it right, and that we need to do it together.
Yeah, I mean, I would say about the first question about patients wanting a clear, certain yes or no, that of course everybody wants that certainty, but actually...
No such thing.
It doesn't exist. It's all murky. We get it wrong all the time. And my experience is that patients also really value honesty about uncertainty and telling people how hard it is to be sure. And it's actually a very good way of building trust with patients to be open about those bits. Otherwise, it's a bit paternalistic to feel like we need to protect patients from, being honest about that. And the other thing I worry about with paternalism is what you were saying about the treatment benefits. I think a lot of us have very heated debates about what is a meaningful benefit. Most of the patients I see in clinic would bite my hand off for the benefit that we've shown in the donanemab and lecanemab studies. 6 months less progression over 18 months, or if you spin it a different way, 50% of patients with no detectable difference at the end of the trial. I mean, it depends on how you look at it, and I don't think we should make too many strong judgments about that. I think we should be open with people and let them decide what's worth it for them.
And I think if I can comment as well, I think it's, as you said, managing patient expectations. And as Cath said, I think absolutely this is about communicating in a way maybe we're not used to. And it really strikes me that if you only go back a couple of years, I sat in a lot more meetings where we were talking about, do we want to give biomarker information when there are no treatments? Again, this very paternalistic view that we didn't have a conversation as to whether people wanted those. We just assumed. But, I sat in meetings with clinical colleagues recently, locally, where we were talking about how potentially could we roll out disease-modifying drugs when they're available on the NHS. And again, there was a lot of skepticism about the actual real-world clinical benefit, and therefore maybe we shouldn't make these accessible. And again, it comes back, I think, to us as clinicians looking at ourselves and saying, we're going to need to have these hard conversations. And I think we should be having these conversations, whether it's with our patients, but also actually, I think, with the general public. I think we need to be making much more about the huge changes that are taking place and the huge opportunities we have so that we can start planning about what our services need to look like in 5 years, in 10 years. So there's a lot of communication that needs to happen. I think there was another question at the back.
I just wanted to follow up on your comments about paternalism. And do you think we're a bit paternalistic about risk in trials as well? Or do you think we've got the balance right? I guess I was on an oncology ward the other day, and they have whole cohorts of patients who are being managed for the side effects of treatments that I think would probably put us off as dementia doctors. And do you think that's a barrier to getting faster trials of more effective treatments?
Yes. I think giving risperidone to someone with dementia is far more harmful than the new treatments we're agonizing about, and you see it happening all the time. And I think that it's not just the risks of treatment, it's risks of other aspects of study participation that ethics committees and R&D departments get really exercised about beyond all kind of proportionality. So people being approached by the wrong person to take part in the study, and what happens if it's not a member of the direct care team? Do people really care if somebody who's not their doctor says, would you like to take part in the study? And we're very exercised about lots of these sorts of things in a way that holds up governance approvals and makes it harder to do research. And we don't really spend the time to find out whether patients actually care.
I think you're absolutely right, yes. I mean, one of the most paternalistic things that's happened recently is that we can't give the drugs anyway, but even if we could, they were not approved in homozygotes for E4. Why are we not giving the information to an individual so that they can make a shared decision with us? I think it's borderline unethical, to be honest. It makes me very angry. So yes, I think that there is a lot that we could do better. The part of the problem is, I suppose, there's an abundance of caution always, partly because there's a partial understanding, back to Tim's point earlier. So the reason we monitor with MRI so often, the reason we stop things when we do, is not because of evidence, it's because that was the way we set it up at the beginning, because we were worried about things happening. The study that was supposed to happen to look at what happens if you actually dose through that symptomatic mild ARIA was set up and then was not done because aducanumab actually got pulled. They were going to do the study. So I think there's lots of things that don't get done that could get done that would, as you say, accelerate things.
Any final comments from the panel on that question? If not, I think we're going to wrap up. Paresh?
Yeah, just one quick comment to that. I think in the same way that different patients and families have different levels of risk they're going to tolerate, I think different physicians and doctors have a tension between us. And I don't agree 100% with what's just been said, because I'm very conservative with a small c, and I tend to sit on the fence a lot as well. So I think having a group of people with different opinions and being able to appreciate that tension too gets us to understand why the things are the way they are, and we sort of move forward incrementally. Is that a fair way of putting it? Yeah, that'll do it.
It's a very balanced way, I think, there, Paresh. Thank you. So on that good note, thank you again for our panel, for all the talks, for all the discussion, and if we can give our appreciation.
Well, I won't keep you long. You've been very patient, and I hope you've enjoyed the meeting. I would just like to say thank you for such high quality presentations. I think all our speakers have been absolutely amazing. And I think the flash talks have been, I could say, even better actually. The discipline, the speed, the information content was absolutely wonderful. And there's been a real buzz about the conference. It's been so good to see you being so convivial, so interested in what's going on. And thank you for all of you that have come up to me and the rest of the team thanking us for this event. So again, you've been a fantastic audience. I'd just like to briefly thank the background team who've put this on. So there's Simon, Rod and Niall and many others. Let's maybe just show our appreciation to them. Yeah. And of course, thank you to our sponsors. So I'm extremely grateful to them. So on Monday, I start writing the DPUK renewal. Okay, now that the conference is sorted, I start writing this renewal. And sometimes people ask, well, what is DPUK really all about? Well, actually, secretly, subversively, it's about change. And if you look at the DPUK agenda over the years, basically, we've been gently changing the structures of the ecosystem to make things more appropriate. So, for example, our data portal, our trials delivery framework, the experimental medicine incubator. So the question is, where do we go from here? And what will I start writing about on Monday? Well, you may remember the very beginning of this conference, I had this strapline, the future is. Well, let me just go through those points briefly again. The future is providing core data services for the people who need them. Do you need a trusted research environment? They are horridly expensive, okay? They take a long time to set up. They take a lot of resource to maintain. Actually, through our Swansea team, we've done it. So you are very welcome to say, come, can you provide a TRE for us? And that would be our great pleasure to do so. It probably wouldn't be cost neutral, but nevertheless, it would be much quicker, much cheaper, and at much lower risk than if you were to do it yourself. So we want to provide core data services, and that means globally as well as nationally. We really do think the future is linking molecular data to health data, both in terms of population cohorts and electronic health records. And we are setting up that pipeline for DPUK3. So if you, again, if this is something of interest to you, if you think this will address questions that could not currently be asked, and there are questions that you are interested in, whether you're pharma, whether you're biotech, whether you're academics, we would be very, very pleased to talk to you. We think the future is a virtual national trials register. And there are several trials registers. Paresh has mentioned one or two, Vanessa and Ivan run one or two. But what we really need is a UK resource which has common materials, common standards, common agreements, common core data sets. And DPUK, as part of its DPUK3, is going to be running pilot projects to establish this, and then using our informatics to be able to link them together, the different registers together. And then we come to what has been called experimental medicine. We've had some great examples of that over the last 2 days. The challenge is, it's a bit like Paresh and the platform trial. You know, you go and you do a grant and your application and you're successful, and it's very, very complicated technology. And then when the grant's over, the technology and the expertise dissipates. What we'd like to do is create core resources, bringing technologies together to create critical mass so that you can address complex precision questions quicker and faster. So that's our agenda over the next few years. As I say, the hard work starts on Monday. And I'm very, very serious. If there is an idea that you have, if there's a contribution that you want to make, then literally now is the time to come and tell us about it. So who do you want to tell? Well, there's obviously James Rowe for experimental medicine. There's Vanessa and Ivan for our trust delivery framework. And there's our Swansea team for the clever data science bit. And also for the data curation, the data processing, there's Sarah Baumeister based in Oxford. So there we are. The door is open for business. We'd very much like to hear from you. And finally, these are our partners, and they've been so good and helpful to us. So thank you to them, and thank you to our sponsors. Have a lovely weekend.